Good morning, everyone. Indira Gandhi Center for Human Ecology, Environmental and Population Studies, University of Rajasthan, Jaipur, in association with International Academy of Physical Sciences, Prayagraj, welcomes you for International Conference on Ecosystem Restoration and Environmental Sustainability, Concerns and Alternatives. Today, we have invited Dr. N.P. Singh, who is retired professor of zoology from the University of Rajasthan, Jaipur, with vast experience of 35 years. He was the head of the department for 3 years and did his post-graduation in zoology with specialization in entomology in 1975 from Aligarh Muslim University. He did his PhD in insect pest management and has been engaged in the studies of spiders. He has credit of publications of about 80 papers in renowned journals and magazines. He has written 4 books on entomology and has guided 26 PhD research scholars. Presently, he is working on the study of spider biodiversity and usefulness of spiders in the pest management. We welcome you, sir, and would request you to start the session.
Okay, thank you. Good morning, everyone. Okay, thank you for this nice introduction. Good morning to all. Particularly Professor Mattu Saab, Mattu. Okay, thank you, sir. So, as already told, I'm talking about this spiders. They play an important role in ecosystem. You know, okay, so I'm just starting introductory part. The spiders are most diverse arthropod group comprising about 47,868 species worldwide. There may be more now because the data belong to 2 or 3 years back. It belongs to 4,114 genera and 180 families. The spiders are cosmopolitan in nature, from the peak of high mountain ranges to the depth of the largest lavines and potholes, from deep marsh to the dry deserts. The spider species are disappearing continuously at an unprecedented rate due to degradation or destruction of specific species habitat by anthropogenic activities. To ensure the expected conservation of biodiversity, the distribution of species needs to be accurately characterized, and this can be achieved only by a complete documentation of the species found in definite area. Thus, a pressing need exists to explore and documenting diversity of spider fauna in Jaipur region of Rajasthan. Simply, the biodiversity is the degree of variation of life forms within a given ecosystem or biome or an entire planet. Biodiversity is a measure of the health of ecosystem. Biodiversity is a part of function of climate. This is the classification: Animalia, Arthropoda, Felicitata, and they belong to class Arachnida and the order Araneae. As regards their morphological part, their abdomens bear appendages that have been modified into spinnerets that extrude silk from up to 6 types of glands. The spiders use a wide range of strategies to capture prey, trapping in sticky web, lacing it with sticky molars, mixing with the prey to avoid detection, or running down, and the most detect prey mainly by sensing vibrations. For the activity, the hunters have acute vision. So, generally, the spiders are carnivorous. Recently, in 2008, one species known as Bagheera sticklingi has been described, which is supposed to be a 90% herbivorous, and sometimes it consumes supposed to be 10% carnivorous. This spider is the only vegetarian species among nearly 46,000 species of spiders, all known to be carnivorous predators. True, the diet of the beekeeping is only about 90% vegetarian, as it sometimes include ants, larvae, and flies. But the researchers say this is the first report of spider that feeds primarily on deliberately on plants, and generally feeds on the acacia leaves, considering that they contain a lot of its proteins. Spider guts are too narrow to take solids, and they liquefy their food by flooding it with digestive enzymes and grinding it with the base edge of their pedipalp, as they don't have the true jaws. The male spiders identify themselves by a variety of complicated courtship rituals to avoid being eaten by the female. So particularly the very good example of peacock spider, but this doesn't mean work in laptop. In computer, however, it is quite easy. The females weave silk egg cases, each of which may contain hundreds of eggs. Females of many species are for the young, for example, by carrying them around or by sharing food with them. A minority of species are social, building communal webs that may house anywhere from a few to 50,000 individuals. The social behavior ranges from precarious toleration, as in the widow spiders, to cooperative hunting and food sharing. Although most spiders live for at almost 2 years, tarantulas and other megalomorph spiders can live up to 25 years in captivity. While the venom of a few species is dangerous to humans, scientists are now researching the use of spider venom in medicine as in place of polluting pesticides. Spider silk provides a combination of lightness, strength, and elasticity. It is superior to that of synthetic material. In spider silk, genes have been inserted into the mammals and plants to see if they can be used as the factories. As a result of their wide range of behavior, spiders have become common symbols in art and mythology, symbolizing various combinations of species, cruelty, and creative powers. In abnormal spider is called arachnophobia. There are certain amazing facts about spiders. The spider silk is the 5 times stronger than silk. Human organs can be created in labs from spider silk. Jumping spider nurse their spider links on spider milk. Spiders are no longer afraid of light. New genus of tarantula is recorded in the Kerala. Nephila comathi, the world's biggest spider, is about 5 inch, 3 feet large breath are there. There are certain myths about spiders. Don't believe everything you hear. The spiders will suck your blood. Yes. When a spider bites you, it doesn't want to suck your blood. It is a warning to you to stay away, and it does this by punching holes in your skin. Tarantulas can kill you, not others. The all spiders spin webs. This is also not true. Actually, a good number of spiders don't spin webs at all. The spiders, jumping spiders, and tarantulas, there are some examples. Some of those spiders that don't spin webs can still produce webbing, but they use it solely to protect their territory. The spiders will bite you for no particular reason. They can bite only if they are hidden inside your shirt or clothing. The daddy long legs are the most poisonous spiders in the world, but they can't bite you. No one is sure how this rumor started. Harvestmen don't have poison. Cellar spiders, which are sometimes called daddy long legs by mistake, have a long amount of weak poison, but only strong enough to make the bite sting a little. What spiders do for you? Don't respect that spider, because the people say are so afraid of spiders that their first reaction is to smash them. If we don't smash them, spiders are one of the miniature best forms of pest control. Many of the other insects you hate, mosquitoes, flies, ants, ticks, and for example, these are typical lunch for these spiders. So next time you feel like smelling, consider number of other bugs you will have to put up with. Spiders kill other spiders also, besides their hunting. The worldwide spiders are carnivorous and dominate on invertebrate predators of insects and other arthropods in every ecosystem in the world. Spiders are the most significant controlling agent which prevent insect numbers reaching alarming proportions, especially pests. In nature, amongst the biotic agency, spiders play a major role in keeping the pest population under control, particularly in the forest ecosystem, where insecticidal pollution is least or nil. Not possible to spray insecticide in this vast forest. In there, spiders have been found to manage the forest pest population to an appreciable extent. The vast majority of spiders are harmless. Sometimes, if provoked, they can leave a nasty bite, but usually they just run away. Only a select few spiders are deadly to humans. Spiders are necessary to the environment. When too many spiders get killed, nature gets thrown out of whack. To do the environment a favor, leave the spiders alone. Don't kill them. So only actually this is some part of a PhD program of one of my students. So that's why in the nearby area of Jaipur, we selected few particular areas. This is one of the marshy area where the selection was made, and this is the woodland area, forest park there near Jaipur. And this is the rocky area, some stones like there, because the some stars are found below the stones also. The spider species found in different habitats of the experimental region were collected and identified after detailed examination. Our collection represented 45 species belonging to 32 genera and 19 families. Now, one by one, I will show you the diagrams along with some knowledge or introduction to them. This is Ocobius putus. This is common name is ant-eating spider or hunting spider, size about 2 mm to 2.5 mm. They are found in corners of houses. The predation method is hunting spiders, ant-eating spiders. Identification, they are very tiny spines, most commonly found in corners of houses. And special feature is that they make small web hide in dent of walls or in the corners. That is the Stegodaphos carcinorum. It is a social spider because they are found in groups and their size about 10 to 12 millimeter. Major habitat is the Acacia nilotica tree and then the thorny bushes generally they are found. And the predation method, their webs are too sticky and compactly spun not to escape allow the prey which happen entangled in the web. The identification they are found in the like weaving colony. The webs of big size and many spiders make a colony in a single web. And this is Stegodaphos species. You could not be identified. The genus is Stegodaphos. It is also social spider, more or less the same size, same habitat. And this is the Uloborus species without venom spider. The feather-footed spider, size 54 millimeter. Their geometric webs on foliage and bark. Predation method, they wrap prey in lots of silk. The cephalothorax is oval, longer than wide, eyes 8, and legs in folding, tarsus 4, more than half as long as the metatarsus 4. The delicate spiders very much camouflage with the background. So here is one male in dark color, female is slightly light color. And this is the Hersilia. They are hunting spiders. They are two-tailed spiders also, size 13.5 millimeter. Then found in tree trunk. The predation method is by hunting the spiders. They found no webs. Two-tailed like spinners are there, long legs. They are very fast moving and they well camouflaged in the background. You see here in the picture also. This is Artima atlanta, also known as commonly known as daddy long legs, because of their very long legs in comparison to the body size. They may be found in the corners of dark places in the houses. They prey upon midges and the mosquitoes. They build irregular web in corners of houses on caves. In identification with long legs, and black joints are seen. You see these black joints are there in the leg. Their body is vibrating and they become invisible when disturbed. So it is a defense strategy of this spider. Basically, Folcus phalangoids, commonly known as also long-bodied cellar spiders. Their size about 8 to 12 millimeter, have very dark corners, tilting, hollow trunks, medium to small size. They fit throughout the soil, cutting of the rocks. They feed on midges, mosquitoes, flies, etc. And the predation method is they build a regular web in corners of houses in the cave. And the identification is body elongated, thin and delicate. And the special feature is their burrowing habits. This is Myrmecinae species, also known as ant-living spiders, because they look like ants in general appearance. They are short bodies. They are only found near ants group. Predation method, they feed on ants and small beetles. Identification is ant-like. Special feature is the found near ant groups. This is also Myrmecinae species, second one. They are also ant-living spiders, short bodies. They are also found near the group. This species is another one. Here this is the Plexippus tychuni. They are domestic jumping spiders of size about 1 inch. They are house dwelling and tree trunks. Predation, they are large house by flying, the broad body with dark black interocular area and a typical white band running from the edge to the posterior eyes near the margin joint of the equally wide. Special feature is the egg sac is guarded by the mother in a silken chamber built at a sheltered corner. This is Chalamonia vittata. They are known as red jumping spider. You see this in front view of this mouth, the head region. The size is 9 to 11 millimeter. The habitat is foliage dwelling. The predation method captures prey by direct attack. They feed on maggots, aphids, etc. And they can identify the body color is red with white or yellow bands. Abdomen is longer than white. Eyes are black in color. It is a beautiful and foliage dwelling spider. That is
मुझे थोड़ा सा ड्यूरेशन बड़ा हो तो इसकी जल्दी
hello.
पहले नॉइज पोल्यूशन में पड़ा है नहीं चीज
नहीं वो ऐसे जैसे मैच होता है उसमें बहुत सारे
जैसे रॉकेट है
Hello. I'm audible.
Yes, sir, you are audible.
Okay. Next is this Thomisa projectus. It is known as the crab spider because the appearance of simple crab. Size is 9 to 11 millimeter. Habitat is the foliage and flowers. The predation method is they feed by direct attack. Never seen spilling any or thread cover to overpower their prey. Then they feed upon honeybees and wasps, etc. So the cephalothorax is quite low but raised near the middle. They show a remarkable degree of camouflage or mimicry. This is the specialty. This one is Chimerus species, a turinode spider, commonly known as, size about 5 to 7 millimeter. Habitat is the foliage, prey is small insects. They are arboreal feeders and feed on insects visiting tender leaves of shrubs. The legs long, abdomen is high, pointed and broader behind. Next one is Distichus minutus. This is woodland crab spider, just like the appearance of the crab. Size about 5 to 10 millimeter. They are found under the bark and on forest floor under leaves and decaying logs. They generally feed on moss and butterflies. The first pair of legs is large and powerful and they covered with many tiny spurs. You see in the picture. This is Heteropoda species, commonly known as huntsman spider, size about 1 inch. They are found in houses, barns, sheds, other sheltered areas, and then they chase their prey by great speed in strong chelicerae used to capture the prey. Identification is by the large brown spider with a flattened body structure and very little dorsal pattern. This is Carinthium, leaf roll spider, size 59 millimeter. They are found in tall grasses. They feed on some hymenopterous insects coliprons and orthoprons, grasshoppers and their limbs. They prey by capture in a web. The size is small, the thorax is wider, and the cephalic region is slightly high and moderately convex. Ocular quite wider than long. See this Fuchsia viridina. They are known as green winged spider. You see this white structure. This is the egg sac. It contains a lot of eggs from which the spiderling they hatch out. Sometimes they tear their egg sacs like this. Large trees, bushes, grasses, feeding method is they feed on small and medium sized arthropods by direct attack. And identification is the long legs and densely covered with long black spines. Abdomen is longer than wide, pointed and posterior portion. Here, this is Octopus creta. This is the most common species in this area. And this lynx spider, they found in grass and foliage of trees and shrubs. They feed on small insects. The predation method is they chase it, they prey rapidly over grass and the foliage of trees and shrubs. They have very long spines found on the legs and 4 rows of eyes of 2 eyes each. This is Oxyopus species, another species also known as lynx spider, size about 1 to 5 millimeter. Habitat, their foliage habitat, foliage dwelling, spine-like habitat. They very actively feeding on the termite swarms. They feed on termites. This is Argyroids species. They are known as mercury spider because of their shining habit. They are parasite. They are found in the webs of dome web spiders, Certophora species. They are found in the web of another species of spider, that is the Certophora species. So they are known as cryptoparasite, and this habit is the cryptoparasite, the stealing prey of other spiders. Sometimes it steal snares of web also. The male silver color and the female is dark chocolate color. And the special feature is the kleptoparasite because they steal prey from the other spiders. Next one is Agilena species. They are known as funnel web spiders. The spider is also known as cobweb spider, size 10 to 12 millimeter. They are found in grasses, bushes, and tree habitats. They generally feed on beetles, hurtling, moths, and small butterflies. They capture in the way. They form web in which they prey are entangled. They capture them. Thorax is wide and has long and hairy legs. This is Pardosa sumatrana. They are also known as wolf spider, size about 67 millimeter. They are found near water bodies and wetlands. Whenever these aquatic insects they come out over the water surface, they are caught by them and they are preyed upon. Hunting spiders which chase their prey. The cephalothorax is brown with tail mediation and 2 lateral tail bands extending from anterior to the posterior margin. The breeding season, the female carry eggs cocoons between the hind legs. This is Hippasa agelinoides, also known as wolf spider, size about 32 millimeter. Habitat is damp area near streams and pools. They prey upon small insects. So hunting spiders which chase their prey. And the cephalothorax and legs are light brown and abdomen is reddish brown with black spots. This is identification mark. And you see this Hippasa species, also wolf spider. In this case also, you see this egg sac. It contains hundreds of eggs. They are found in grasslands, damp or near swimming pools. The predation is they feed small insects by direct attack. Identification mark is that the cephalothorax and legs are yellowish brown and abdomen with greenish brown patches. They are actually moving spiders and found in groups. This is Lycosa species, also known as wolf spider. They are found near the streams and aquatic bodies in isolated areas. There they feed upon small polyprons, small butterflies, moths, fruit flies, aphids, plant hoppers, their nymphs and adults. They are expert ground-dwelling hunters. You see here the Cyclosa species. It is also known as a signature spider because the web formed by it is similar to the signature of someone. It's, we couldn't record its habitat, but this signature was this type of found in the bushage area. They kept the prey in the web, abdomen were wide and the caudal project at the hind end bent conical. They make signature-like structure in the web. You see, Neoscona, they are known as orb web weavers. They are found in moist woodlands. They prey upon aphids, beetles, flies, wasps, thrips, small moths, small butterflies, and the previous method they capture the orb web. Orb web means the web which are formed by a large number of linear web-like structures radiating from the center. And so once the capture is entangled, it is unable to get free. Ultimately, they consume them. The females reddish brown to brown, males light brown, with the sides of the carapace nearly black. They make beautiful orb webs. This is Dioscona They are also orb web weaver, more or less same like Dioscona, this one. This is Dioscona pavida, also orb web weaver. They are all kind of habitat they are found. They weave orb web. The cephalothorax is longer than the wide and narrow in front and clothed with fine fibrous-like structures. This is Neoscona mukherjee. This is orb weaving spider, size about 12 millimeter, a tall grasshopper and bushes. The small butterflies, for fruit flies, aphids, plant hoppers, nymphs, and adults, they are consumed by them. They spin a thin threaded small sized radial geometrical web. Abdomen is sublingual, longer than wide, tapering in the posteriorly and dorsum with a club-shaped grayish-white patch. You see this one, Jagila melanocorata. They are also orb weavers, size 1 to 3 inches. They are habitat green plants and trees, and they are reproduction method by capturing in the huge web. They are big size spiders, short cephalothorax and oval-shaped abdomen. They are large specific web structure. They are known as Setophora citricola. This is also one of the abundant species found in Jaipur region. Their size about 11 to 18 millimeters. They are found in dry areas. Prefer Euphorbia, this strain, for making dome web. They are distributed throughout India. The predation method is by making waves, dome web. The abdomen thickly covered with pubescence and hairs. You see here the this is this huge colony of Cytophora sativa. This is a tree in this nearby area in Rajasthan, Jaipur, and this is acacia tree. The whole tree has been covered by the huge colony of Cytophora. You see this white web-like structure. And this is Cytophora psychotrosa. The 10th orb web spiders. Their size 69 millimeter. They are found in dry area. They prefer Euphorbia strains for making dome web. They prey upon small butterflies, moths, fruit flies, aphids, plant hoppers, their nymphs and adults. They entangle the prey in the web. A pair of black patches on the lateral longitudinal sides and a single black patch in the middle region of the cephalothorax. This is identification mark. You see this, UK decorata. They are also known as orb web weavers spiders, size about 8 to 9 millimeters. They are found in low shrubs and moist environments. They form slant orb web. The cephalothorax is flat, which is pale yellow, darkish medially and towards the margin. The special feature is that they are most beautiful spiders, so it's the name of the species decorata, UK decorata, because of their beauty. Here is the Caragnatha mandibulata. They are long cylindrical spiders or star spiders, they are known as, size about 2.5 centimeter. They are found near aquatic and damp places. They are small feet feed on small cauliflower, small butterflies, moths, fruit flies, aphids, plantophores, and mixed adults of autochthons. The predation methods by web breathing. The web are simple to 3 cm. Long abdomen and long legs are exceptionally long. In this case, this is Oleos species, also known as huntsman spider, size 8 to 10 millimeter. They are found in house-sized buildings and their carapace is convex. They are very fast-moving spiders. This is the identification mark. You see here, Scytodes species. They are foliage-dwelling spiders. They are found under stones also. You see here also the egg sac is there under their body. The small insect, nocturnal spiders, they spit sticky and poisonous substance over its prey to make it unconscious. And cephalothorax high and subglow and tend to leg. Legs, joints, black in color. Pardosa species, soil lacosa species spiders. They are ground dweller spiders. The carapace convex. They are very fast moving spiders. Also, this is Ogyopus mechanicus, also known as cross-breeding spiders. They are found in grassland and low shrub, size about 10 to 12 millimeter. They are known as hunting spiders also. The carapace is convex in this case. The Echernea species, the rolled leaf spider, they are known as. They are found in low vegetation. They are also hunting spiders. The carapace is convex. The special feature is that the spiders falls to the ground and acts as dead when someone tends to be nearby. They fall to the ground and acts as being dead. This is Hersias adenophony. Adenophony is house jumper. They are known as size 6 to 8 millimeter. They are found in house walls and gardens, and they are hunting spiders. Here is the Rene species with prey. This is the larva of Caterpillars. This is a spider, but nearby. They are found in bushes in small trees. They are hunting spiders. So finally, all these species so far discussed by me, they were purely carnivorous. I told you in the beginning, this is the Bagheera Pipilingi. This is supposed to be the only spider species which is vegetarian rather than carnivorous, and it generally feeds on the small newly formed leaves of acacia, considering them that these leaves contain a lot of protein, and the protein is the main diet. And sometimes they are seen to consume small insects, small aphids, only sometimes, then the acacia leaves may not be as big. So this was the basic account which I wanted to show you, because in all cases you might have seen that the feed on a variety of insect species. That's why they can be used, or they are used to control a large number of insects. There may be few beneficial one also, but mostly the insects which are pests, they are consumed by them. If there's some idea regarding their web species or web, sheet web, net web, funnel web, triangular web, single line, thread web, the other diagram. They are such a benefit to human beings. The main benefit that they are controlling in nature without uh, this activity of human beings. So they can be cultured in the lab. They can be in large amounts after culturing. They can be released into the fields for our purpose. So spider genomes may be a less polluting alternative to conventional pesticides, as they are deadly to insects, but great majority are harmless to vertebrates. So Australian web spiders are a promising source, and most of these insect pests have had no opportunity to develop any immunity to their venom. And funnel web spiders thrive in captivity and are easy to milk. So it may be possible to target specific pests by engineering genes for the production of spider toxins. and virus that infect species such as cotton bollworm. The possible medical use for spider venoms are being investigated for the treatment of cardiac arrhythmia, Alzheimer's disease, strokes, and erectile dysfunction. The peptide GSMPH4 found in the venom of Brachytherma vegans is being researched to determine whether or not they could be effectively be used for the treatment of thalassemia, muscular dystrophy, and myoma. Because of spider, the high, both light and very strong, the cells are being made to produce it in goats, milk, and in the leaves of plants by means of genetic engineering. So spiders can also be used as food. The cooked tarantula spiders are considered a delicacy in Cambodia and by the other countries, and the Indians of southern Venezuela, provided the high retail hairs. The spider's main defense system are removed first. And they generally you see that in our houses, particularly the housewives, when they see any spider, they are just scared of. So this is can be known as arachnophobia. So arachnophobia is a specific phobia. It is the abnormal fear of spiders or anything reminiscent spiders such as webs or spider-like shapes. It is one of the most common species phobia, and some studies show that 50% of the women and 10% of men show symptoms of this arachnophobia. It may be an exaggerated form of an instinctive response that helps the humans to survive, or a cultural phenomenon that most common in predominantly. So spiders in symbolism and in culture. The spiders have been the focus of stories and mythologies of various cultures of centuries. The Italian tarantula is a dance to rid the young woman the lustful effects of a spider bite. The web spinning also caused by association of the spiders in creation myth, as they seem to have the ability to produce their own world. Dream catchers are depictions of spider web. The Moche people of ancient Peru, they worshipped naturally. They placed emphasis on animals and often depicted spiders in their art. So this is some significant study regarding spiders. Okay, this is done now. Thanks. Mhm.
Thank you, sir, for showing us the amazing world of spiders. Now, I would like to welcome our second guest, Professor S.C. Joshi. He is UGC Emeritus Fellow in Center of Advanced Studies from Department of Zoology, University of Rajasthan, Jaipur. He was Dean of Student Welfare and International Students Advisor, Coordinator of College Development Council, Additional Director, Center for Converging Technologies, University of Rajasthan. His research contributions include transdermal drug delivery system, atherosclerosis, and reproductive toxicology. Sir has been felicitated by various awards and gold medals like Lifetime Achievement Award in 2015, Green Globe Award in 2014, Gold Medal by IAES, Haridwar in 2012, and Environmentalist of the Year in 2013. Sir has published more than 200 research papers of national and international repute. Thank you, sir, for joining with us. Now, I would like to request you to address the gathering. Over to you, sir. Sir will be joining us shortly.
In the meantime, we shall like to thank Professor N.P. Singh for giving a very informative lecture on spider diversity. And we are thankful to Professor S.S. Chauhan that during this COVID, we could see each other online through this seminar. So Professor Singh, it was very innovative and very informative lecture, and we also learned many things from you. Thank you so much.
Hello. Namaskar, sir. Sir, thank you so much for such an informative lecture. Hearted congratulations for the immense wealth you have given us by sharing your immense and boundless knowledge. Thank you so much, sir. Meeting you after so many years, as we met in Shimla only, so now meeting online. But still, let's thank God that at least we can meet on this medium also. Thank you, sir. Thank you so much.
हम्म? नहीं आगे आ जाना दूसरी गाने गए बढ़ गया देर लगी अब किससे करें टक्कर वो आगे से जवाब ही नहीं Professor Singh
Having some technical issues at the end of sir, so hopefully you'll join us shortly. Thank you.
Professor Singh
Hello. Yes, dear participants, you can interact with Professor Mattu by the time Dr. Joshi comes, if you want.
Any participant who want to interact with sir, they can raise their hand here. Or they can ask us to unmute themselves.
Professor Singh, can you hear me?
Okay.
It was very interesting talk. One thing you told that spider venom can be used for the cure of Alzheimer's disease. Is there any report? Because
No,
Alzheimer is very important.
it is simply uh, the people are thinking about it in Australia and other countries.
Any report, any paper?
No, report is not there.
Still they are working, because it is
No.
very, very important disease. These days, lot of people are under
Alzheimer's
depression.
disease.
So Alzheimer's disease and memory loss are happening.
And this to be used as pesticide against pests, it is quite possible. Because low dosage.
Secondly, there was data that 50% of women are scared of spider and only 10% of men. I think it is old data. Now, women are not afraid. I think if we conduct survey now, women will be not be far behind men. Because 50% of women, they are afraid of spiders and 10% of men.
Kill in the kitchens and others, they simply, but generally, by habit, they have this tendency to show the scaredness in humans. They generally kill them.
एक और चीज है, घरों में अगर स्पाइडर वेब इकट्ठे हो जाते हैं तो लोग इसको इनऑस्पिशियस मानते हैं. व्हाट कैन बी रीजन?
सफाई आजकल दिवाली पे सबके सफाई हुई है. इवन मैंने भी मारी है.
क्या रीजन होगा कि स्पाइडर वेब को हम इनऑस्पिशियस क्यों मानते हैं? दे आर वर्किंग सो हार्ड टू मेक द वेब, बट वी आर कंसीडरिंग इनऑस्पिशियस.
मान के चलते हैं ना, इनको देखते ही मानने की हिम्मत करते कोशिश करते हैं. ठीक है, और यो मैं खुद कह रहा हूँ कि मत मारो इनको, मगर घरों में हम इनको टॉयलेट नहीं कर सकते. बाहर तो बहुत अच्छी जरूरी है ये,
क्योंकि अदरवाइज वी सी स्पाइडर आर सच हार्ड वर्किंग इंसेक्ट. They are making their webs, and you said that silk thread is more.
Yeah.
Recording in progress.
इनके लिए जो कहा है मिलिट्री के लिए जैकेट, बुलेट प्रूफ जैकेट बनाने का है.
हाँ जी, you said that.
तो देखा पड़ा है मैंने.
But only few scientists now in country are working on spider. There's more should come forward, because spider, you know that only few.
In the beginning, I was also scared कि ये गंदी होती है, कैसे काम करें. और बाद में दिमाग में आया कि बहुत अच्छा काम है. जो लिटरेचर देखा तो लिटरेचर में तो बहुत पहुंची थी जानकारी.
In Shimla we have one scientist, Dr. Chakravarti in HFRI.
लुधियाना में करते थे वहाँ नाम भूल गया उनका.
यहाँ हमारे शिमला में भी HFRI में अब चले गए Dr. Chakravarti from Kolkata. He worked on spiders. He gave me book also. He has written a book on spiders also, diversity. So he worked on the forest spiders.
वो कैंडिडेट जो आपने सेलेक्ट की थी, उसने अपने कैंडिडेट को काम दिया है ये अभी डिपार्टमेंट में जो आपने किया था एसोसिएट प्रोफेसर का. उन्होंने काम आगे बढ़ाने की कोशिश किया है. तो अलग होगा.
डॉक्टर विनोद कुमारी. Very good, because somebody should carry this work forward.
Definitely.
As in our case also, only few scientists are working on honeybees.
Honeybees, right.
Everybody is going after biotechnology these days. So that is the problem with the other branches. Because we have to have a balance between modern science and old science.
अपन तो थोड़े से आउटडेटेड हो गए इस काम के लिए. आप तो अभी बहुत इंगेज्ड हो, दोनों आप लोग मिलकर, मैडम भी और आप भी दोनों मिलकर तो काम भी करते हो. इसलिए मेरे को जब बंदे ने मेरे को कहा सुरेश चौहान ने, तो सबसे पहले मैंने आपका नाम बताया. भाई हमारे को बड़े शिक्षक हो रहे हैं, इनको तुम कांटेक्ट करो जाके.
Dr. Chauhan is a very nice person. He daily talks to me and we are friends, although we have not met yet.
मेरे तो पुराने फ्रेंड हैं, आपके नए फ्रेंड बन गए. अच्छा है, अच्छा आदमी.
Thank you so much, sir. Thank you so much, sir. It was
आपको
really
मैं
being great here. Dr.
आप
Joshi, namaskar.
Namaskar.
Namaskar,
How
Dr.
are you?
okay, good morning,
Yeah,
Joshi साहब.
waiting after a long time.
We are waiting anxiously for you.
Hmm.
So good to see you,
Yeah,
sir. So good.
yeah. Thank
So
you.
all
Thank
the very
you.
best, sir. Best of luck, sir. Okay, sir.
Thank you very much. Thank you, sir.
जोशी जी का लेक्चर तो हमेशा इनोवेटिव होता है. Today
Hmm.
also I think it will be very good.
यूनिक होता है यूनिक जोशी जी.
Hope so. चलिए.
I would like to welcome Professor S.C. Joshi. He is emeritus in Center of Advanced Studies at University of Rajasthan. Sir was Dean of Student Welfare, International Student Advisor, Coordinator of College Development Council, and Additional Director Center of Converging Technologies, University of Rajasthan, Jaipur. His research contributions include transdermal drug delivery system, atherosclerosis, and reproductive toxicology. Sir has been felicitated by various awards and gold medals like Lifetime Achievement Award in 2015, Green Globe Award in 2014, Gold Medal by IAES Haridwar in 2012, and Environmentalist of the Year in 2003. Sir has published more than 200 research papers of national and international repute. Thank you, sir, for joining with us. Now, I would like to request you to address the gathering. Over to you, sir.
Thank you. Thank you for a nice introduction. Just start करते हैं. Okay, thank you very much for nice introduction. And today I will speak on heart disease and factors, risk factors, and preventive role of the Indian Passage. Friends, you know that this heart disease is a very, very big problem throughout the world, and many people they are suffering from this disease. And now the world has accepted that Indian diets and the Indian spices, they are good. They are very good for prevention of the heart disease. So here I would like to mention about some Indian spices. They have potential role in prevention of heart disease. So first of all, I would like to say what is atherosclerosis. This atherosclerosis is a combination of changes in the intima of arteries consisting of focal accumulation of lipids, complex carbohydrates, blood and blood products, and also associated with medial changes. So friends, this atherosclerosis, an insidious and dangerous disease, a progressive chemical and structural injury to blood vessels in such critical organs such as the heart, brain, and kidney. So friends, this atherosclerosis not only occurs in the heart, but it also occurs in the brain and kidney and other organs also. The hallmark feature of atherosclerosis is the buildup of cholesterol into lesions called plaque that can reduce the flow of blood. The deposition could be of fatty substances, cholesterol, cellular waste products, calcium, and fibrin in the inner lining of the carotid arteries, causing stroke, and in coronary arteries, causing heart attack. So these are the different terms. We can understand what do we mean by stroke, what is heart attack, and when the delivery of blood to heart muscle drops enough, causes angina, and in lower extremities causes intermittent claudication. And the plaque may rupture, stimulating the development of thrombus. A large thrombus on the surface of a ruptured plaque can cause obstruction of the lumen of the artery. So these are the various terms related to heart disease like stroke, heart attack, angina, intermittent claudication, myocardial infarctions. Then if we talk about the risk factors of the atherosclerosis or the heart disease, so we can classify the risk factors into two categories: the factors which can be controlled or modifiable, or the factors which cannot be altered or which cannot be controlled or which are non-modifiable. If we talk about the factors which can be controlled, our diet, we can control the diet. Hyperlipidemia, we can control the hyperlipidemia by exercise, by taking some medicines, Oxidative stress, we can reduce the oxidative stress. Diabetes mellitus, we can control through medication and lifestyle changes. We can control hypertension, endocrine disorders, physical activity, metabolic activity, smoking, obesity, depression, and immunity. So these are some risk factors which can be controlled. But there are some factors we cannot control them, or we cannot alter them. For example, age, we cannot change the age. Sex, no, we cannot change the sex. Then family history, we cannot change the family history. So these factors, if the atherosclerosis is related to age, sex, or family history, then it is little bit more dangerous. So if we think about what is the mechanism of atherosclerosis, then very basic fundamental mechanism of the atherosclerosis is atherosclerosis is initiated by oxidation of LDL in blood. Then oxidative modification of LDL results in the migration of certain white blood cells from blood into the artery wall. Then monocytes are converted into macrophages, which essentially sequester or take up the oxidized LDL from foam cells in artery wall, and then aggregation and accumulation of foam cells leads to fatty acid formation. Then smooth muscle cells migrate from media to the subendothelial space, where they proliferate and produce connective tissue to form a fibrous plate. Fibrous plaques are soon covered by a thick dome of the connective tissue with embedded smooth muscle cells that usually overlay a core of lipid and necrotic debris. And finally, growth of the fibrous plaque results in vascular remodeling, progressive luminal narrowing, blood flow abnormalities, and compromised oxygen supply to the target organs. So this is how this atherosclerosis occurs. This is the simplest mechanism. You can see here the first picture is control aorta. This aorta consists of tunica intima, tunica media, and tunica interna or adventitia. And the second picture is showing the atherosclerotic plaque. These are the plaques. You can see the plaques in the aorta. This is the diagrammatic representation of the endothelial dysfunction atherosclerosis. So this you can see here, all the three layers are there, and how leukocyte migrations occur. And because leukocyte migration occurs due to changes in the endocrine permeability, and these leukocytes or monocytes, they attach to the endothelium, and you can see how these are attached. This picture shows how the smooth muscle cells, they migrate, form cells, how they form cells are formed, and T cell activation and different activities of the, you can see here the plaque is forming. Next, mature or advanced plaque is here. You can see rupture of the plaque. If foam cells, they causes pressure on the endothelial wall, then endothelial wall rupture and blood comes out. So this is very critical condition in the development. Then if we talk about the oxidative stress and atherosclerosis, so because atherosclerosis is very much directly related to oxidative stress, so oxidative stress may cause development and progression of the atherosclerosis. It can be defined as a state of disturbance in the pro-oxidant or antioxidant balance in favor of former, leading to potential damage. The process associated with oxidative stress involve reactive oxygen species and radicals that can result in elevated levels of oxidatively modified or toxic molecules that finally lead to the formation of foam cells from which atherosclerotic plaque evolves. Most living species have protective system against oxidative stress, like antioxidants synthesized within the body or taken in the diet that counteract oxidative damage by reacting with free radicals and protect lipoproteins from oxidation. These antioxidants are enzymes, many vitamins and minerals that provide powerful protection for the body. And because now I will discuss how this oxidative stress causes atherosclerosis and how this oxidative stress can be reduced by using Indian spices. Then a large number of drugs have been used for the curing of the hyperlipidemia or atherosclerosis, like bile acid sequestrant, fibric acid derivatives, HMG coenzyme reductase, nicotinic acid, SL coenzyme, probicol, azimite, HTMI. These are the various synthetic drugs which are used for the curing of the atherosclerosis. And the treatment with antioxidants, minerals, omega-3 fatty acids, dietary protein, and dietary fibers are also beneficial in prevention of atherosclerosis. If we talk about the Indian spices, now we come to the Indian spices. And Indian spices, you know, they are esoteric food adjuncts that have been used as flavoring and coloring agents, as preservative for thousands of years. Spices have also been recognized to possess medicinal properties, and their use in traditional synthesis of the medicine has been on record for a long time. With the advancement in the technology of spices and on knowledge of the chemistry and pharmacology of their active principles, their health benefits, effects were investigated more thoroughly in the recent decades. Many health benefits attributes of these common food adjuncts have been recognized in the past few decades by pioneering experimental research involving both animal studies and human trials. These studies documented digestive stimulant action, hypolipidemic effects, anti-diabetic influences, anti-lithogenic property, antioxidant potential, anti-inflammatory property, anti-mutagenic property, and anti-carcinogenic potential of spices. Among these, the hypocholesterinemic and antioxidant properties of few specific spices have far reaching nutraceutical values. So I will speak about some Indian spices. We have done a lot of work on Indian spices and other herbal products on prevention of the atherosclerosis or hyperlipidemia. I will show some these Indian spices which are very useful in prevention of the atherosclerosis. For example, this allium sativa, commonly known as garlic, and it is a herb that has a lot of medicinal usage, protective effects of organ sulfur compounds from garlic on atherosclerosis have been attributed to its capacity to reduce lipid contents of arterial wall. Garlic contains sulfur containing compound allicin. So allicin is the allicin or which is converted into active ingredient allicin. So allicin is the active constituent of the garlic, and this allicin is responsible for the reduction of hyperlipidemia. Just a minute, some problem with the computer. आगे नहीं बढ़ रहा है. So this is the structure of allicin. Next Indian spice is cinnamon verum or cinnamon. The cinnamon oil has had positive effect on antioxidant metabolism. Besides, increase the antioxidant enzyme activity and decrease the serum MDA levels. Phenolic compounds such as hydroxycinnamaldehyde and hydroxycinnamic acid present in the cinnamon extract scavengers of the peroxide radicals and prevent oxidative damage. In addition to the effect of cinnamate, a phenolic compound found in cinnamon bark and other plant metals on lipid metabolism, antioxidant enzymes activity in rats fed a high cholesterol diet has been studied and indicated that cinnamon suppresses lipid peroxidation via the enhancement of hepatic antioxidant enzyme activities. So these are the three active compounds present in the cinnamon. Then Commiphora mucul, commonly known as gugul. Gugul is used since ancient time, and these are the active components of the gugul are gugulosterone E and gugulosterone Z. So two types of steroids are present in the gugul. Coriander sativum, locally known as dhania, and dhania is also very effective in reducing the lipid or cholesterol. It has many active compounds like caffeic acid, chlorogenic acid, quercetin, camphorol, remitin, and epigenin. So these are some flavonoids are there. Flavonoids, isoflavonoids are there, and these are very this coriander is very good antioxidant. Curcuma lunga, haldi, turmeric, commonly known as, and you all know about the constant of turmeric, and it contains bis-methoxycurcumin, dimethoxycurcumin, and curcumin. These are three active constituents found in the uh, curcuma longa, and they are very effective in reducing the blood lipid levels. Myristica fragrans, nutmeg, locally known as jaifal, and this jaifal is also very potential antioxidant agent, and this jaifal contains eugenol, isoeugenol, and methoxyeugenol. So these are the three important active constant of this jaifal, which prevent the atherosclerosis. Amomum subulatum, cardamom, and this Amomum subulatum is also very important spices, and it contains various active compounds, and these, like for example, petunidine, leucosamidine, subulin, and cineol. So different, they are isomers of this Amomum subulatum compounds. So here I would like to discuss about the details of Amomum subulatum. We have worked on detail with all these Indian spices, but here I would like to present some findings of our laboratory, how Amomum subulatum is effective in reducing the atherosclerosis or hyperlipidemia or oxidative stress. So I'll show some pictures. So 70% methanolic extract, crude extract of subulatum was used for the this experiment. Animal, we conducted our research in rabbits, and various groups were prepared for were designed to conduct the experiment. And for example, we use control group, placebo control, then we fed them cholesterol for 60 days, then we fed cholesterol for 120 days, then we sometimes we withdraw cholesterol, we gave cholesterol plus this compound from day 1 to 120 days. In some experiments from 6 after developing cholesteremia, then 160 to 120 days. So different groups were prepared, and we analyze blood total erythrocyte count, TLC, DLC, hemoglobin, hematocrit, blood sugar, blood urea, for just normal to assess whether they have toxic effect or not, whether this compound, this Amomum subulatum, has any effect on TLC, DLC, hemoglobin, hematocrit, blood sugar, and blood urea level. And in serum, we estimated total cholesterol, triglyceride, phospholipid, HDL cholesterol, LDL cholesterol, VLDL cholesterol, LDL ratio, and different ratios were prepared. Then in tissue, we also conducted cholesterol, phospholipid, triglyceride estimation in liver, heart, aorta, kidney, and coronary artery. And if we talk about the enzymes, then we conducted lipid peroxidation, catalase, glutathione estimation in the liver, heart, and the aorta of the rabbit. We also performed some planimetric study. We measure the diameter or different layers of the aorta, and fecal biochemistry was also conducted for cholesterol and phospholipid estimation, and in liver, heart, aorta, coronary artery, kidney, histology was also studied. So there was no changes in hematology was observed. You can see there are some important changes in serum total cholesterol and triglycerides after feeding cholesterol. You can see this yellow bar. Cholesterol level and triglyceride levels were increased after cholesterol feeding, and after different dosage of the amoxicillin, the cholesterol was decreased. You can see in all diagrams. Here you can see serum phospholipid was also increased after cholesterol feeding, and this serum HDL cholesterol, and here see serum phospholipid was decreased after treatment with various dosage of the amoxicillin. Here you can see serum LDL. LDL is known as bad cholesterol. So LDL cholesterol was increased and decreased after treatment. Serum VLDL ratio was also, cholesterol was also increased and decreased after treatment. Here you can see HDL ratio. You can here see this HDL ratio was decreased in cholesterol fed rabbit, and after treatment it was increased. So this HDL cholesterol is a very important factor or very important parameter in estimation of this atherosclerotic research. HDL cholesterol, you know, the LDL cholesterol is known as good cholesterol, and it should be increased after treatment, because the heart patients or the patients with hyperlipidemia, they have lower level of the HDL cholesterol and higher level of the LDL and VLDL cholesterol. So the drug should be like that. If the drug or the treatment reduce the LDL and VL and increase the HDL cholesterol, then one should think it's a good drug, or it can prevent oxidative stress. Here you can say this cholesterol in liver was also increased in cholesterol fed rabbits. It was also increased in heart of cholesterol fed rabbit and decreased after treatment. This cholesterol in aorta was also increased. Triglyceride in the liver was also increased. Here you can see this triglyceride in the heart and triglyceride in aorta was also increased in cholesterol fed rabbits and decreased after treatment with the amoxicillin. Here you can see these are the phospholipid in liver, in heart, and aorta. They were increased in cholesterol fed rabbit and decreased after treatment with the various dosage of the amoxicillin. Similarly, this total wall area of it's very important diagnosis in the development of atherosclerosis. You can see the wall area of ascending and thoracic ascending aorta and thoracic aorta, and this abdominal aorta. The aorta wall was thick in cholesterol fed rabbits, and it was thin after treatment with the amoxicillin. Here you can see ascending plaque size. Plaque size was increased in the aorta, all the three aorta, ascending, thoracic, and abdominal aorta, and this plaque size was reduced after treatment with the amoxicillin. Here you can see this lumen size. Here lumen size was decreased after cholesterol feeding in ascending, thoracic, and abdominal aorta, and this lumen size was increased after treatment with the amoxicillin in all the three aorta, whether it is ascending aorta or thoracic or abdominal aorta. It's very important, because after treatment the lumen size is increased, so blood flow increases, and if the lumen size is decreased, then the blood flow stops and it causes myocardial infarction or heart attack, like this thing. Here, fecal biochemistry, fecal excretion was also increased after treatment with the this amoxicillin. And here you can say these are three very important enzymes like lipid peroxidation. So lipid peroxidation was increased in cholesterol fed rabbits, and lipid peroxidation was decreased after feeding with the amoxicillin. Here you can see lipid peroxidation in liver, heart, and aorta. In all three organs, this was increased and decreased after treatment. You can see here this glutathione. Glutathione was increased in cholesterol fed rabbits of this glutathione in all the three aorta. Glutathione heart, liver, and aorta. It was decreased in cholesterol rabbit, and it was increased after treatment with the amoxicillin. Here you can see catalase was also decreased in cholesterol fed rabbit, aorta, heart, and this liver. Yeah, you can see here the histological pictures of the aorta. This you can see all three normal aorta, ascending aorta, thoracic, and abdominal aorta. All three aorta they have tunica, externa, media, and interna, and you can see normal cells of cells are here. You can see after cholesterol feeding, plaques are seen, form cells are seen in all three aorta. You can see plaques, abdominal aorta after 120 days. If it stop the treatment, then plaques gradually, some plaques they reduce, but not cure. After treatment with this amomosulatum, you can see various regression in the plaques. You can see how it works. And you can see almost towards normal size of the plaques. Of course, plaques are there, but towards normal. So it means plaques are regressed after treatment with the amomosulatum. See, plaques are regressed almost towards normal, just normal. After if the dose is increased, it's about 200 milligram per kilo body weight. The structure was almost normal. You can see liver, normal liver after cholesterol feeding, fatty liver. You can see 120 days, 60 days, different days durations. You can see changes in the liver after treatment. Fatty regression is regressed. Fatty accumulation is regressed here. You can see changes in the liver. Kidney, normal kidney, Bowman's capsule glomerulus. You can see some degenerative changes in the kidney here, in glomerulus. After treatment towards normal side, you can see heart muscles normal. You can see changes in the cardiac muscle after 120 days and 60 days treatment cholesterol feeding. Towards normal, almost normal. If we treat them 200 milligram per kilo body weight from day 1 to 126 days. So concurrent feeding means drug and cholesterol both are feeding here. You can see coronary heart artery normal. You can see this lumen size is reduced. See the normal lumen after 200 milligram of treatment, just normal historic texture of the artery. So if we talk about what are our important findings, then we can say that our results indicate that amomosulatum crude extract possesses significant hypolipid activity as indicated by significant lowering of serum and tissue lipid profile. The hypolipid effect of sulatum might be partially due to enhanced fecal loss of cholesterol and phospholipids, and regression in atheroma suggested its anti-atherosclerotic nature, non-significant changes in hematological parameters, advocated non-toxic nature of plant products, reduction in lipid peroxidase measured as TBRS activity indicated its anti-lipo peroxidative nature. Amomosulatum had remarkable potential protective action on free radical mediated oxidative stress and restored the GSH and catalase level, thereby accelerating the repair mechanism and significant protective effect. The property of activity of amomosulatum crude extract may be due to its antioxidant enzymes, which provide defense against the detrimental action of the LDL oxidation. And what we can recommend from the above mentioned findings, we can conclude that discriminate and proper use of these Indian spices is safe and may have clinical and public health relevance. However, further research is intended to fully understand the regulation and identify specific compounds from various Indian spices and their products that are responsible for most of its biological effect. So to develop the safe and cost-effective anti-atherosclerotic agent of plant origin. So friends, these are the some findings which and I was able to present in front of you. So any comment, any suggestion?
Namaskar, sir.
Namaste, namaste, madam. Just there is some problem in computer. I am just opening it.
Haan ji,
Just a
sir.
minute.
Hello.
Yes, हाँ, नमस्कार madam. Namaste
हाँ
sir.
जी,
Namaste.
हाँ जी, sir. Sir, congratulations for such an enriching lecture. And it's really need of the of an art. All the things we have in our kitchen, our haldi, our turmeric,
Yeah.
the cinnamon, yeah, and the garlic, we all need them badly. And you told very nicely about the significance. Sir, can we know that what quantity should be used, like cinnamon and all? Because we all know that we have to use it, but we don't know actually how much quantity we must use it on daily basis, sir.
Yeah, it's very relevant thing. Just as normal, it depends on your taste how you use, but don't use in excess.
Yes, sir. Yes, sir.
Don't use excess spices in diet
Yeah.
because they may
Yes,
cause another
yes,
complications.
yes, sir.
Just
Thank you so much, sir. Thank you so much. We will implement your all the suggestions right from today.
yeah,
Very
thank you.
strictly.
Thank you.
Sir. If you need to
So how
strengthen
are
our
you?
heart. Yes, sir. Thank you, sir. Thank you so much.
गुड आफ्टरनून प्रोफेसर जोशी.
सर नमस्कार.
नमस्कार. आपको काफी टाइम बाद देख रहे हैं.
अरे so nice of you, great sir.
Dr. Chauhan has enabled us to meet you through online mode.
Yeah.
So you had a very good, wonderful lecture. You always deliver. I have heard you many times. Today also it was very wonderful lecture on heart, and it is very very important topic. यहाँ छोटा सा एक क्वेश्चन मेरा है कि गरम मसाला यूज किया जाए कि नहीं किया जाए?
बिल्कुल करना चाहिए सब, लिटिल क्वांटिटी के अंदर,
क्योंकि पाइल्स की
तो अगर
प्रॉब्लम
ज्यादा अपन काम में लेंगे तो फिर वो अल्सर वगैरह पोजिशंस वगैरह की जाए, तो अपना गरम मसाला इंडिविजुअली आप यूज करें.
जैसे
बना के
अगर आप some has piles, then it creates problem, garam masala.
Yeah.
Other disease also. So some doctors suggest that don't use garam masala.
Yeah. So if you have some complications or ulcer in this or like that pile, so then don't use them.
Otherwise
लेकिन
all
बस ये बताने का मतलब ये है कि सब हम लोगों में अभी भी प्रॉब्लम कम है, क्योंकि हम लोग सदियों से ये काम में लेते आ रहे हैं. तो अगर कंपेयर करें हम वर्ल्ड में थ्रू आउट, तो अभी भी इंडिया के अंदर कंपेरेटिवली जो है वो ये प्रॉब्लम कम है. और इसकी जब रिसर्च, वी हैव डेटा फ्रॉम ऑल ओवर द वर्ल्ड, व्हाई इंडियन पीपल दे हैव स्टिल, आई एम इन कंपैरिजन टू अदर वेस्टर्न एंड other countries, still we have lower incidence of heart disease or prostate cancer or breast cancer, because we use lot of fibers, we use lot of Indian spices, herbal products and plant products, so that's we are little bit safe.
Okay, congratulations Dr. Joshi.
Thank you sir. Namaskar.
नमस्ते. और मजे में हो?
बस सर बढ़िया, everything is fine. चलिए, अच्छा लगा सब मिलके, है ना? सुमन साहब को थैंक्स देंगे, सबको मिला दिया. मटू साहब जल्दी ऑफलाइन भी मिलेंगे हम लोग, वेरी सून.
Thank you.
Thank you so much, sir. I just recall when you came to Shimla. I think we have not met after that. So we wish that conditions be at ease right now, and we can meet each other. Same with Professor Singh also. We met in Shimla also only.
Thank you, sir.
Thank you so much, sir. Thank
So
you.
it appears that it is a conference of zoologists. All
जल्दी मिलेंगे सर, बिल्कुल
जल्दी मिलेंगे बिल्कुल.
Yes
और
sir,
आप
yes sir.
बाकी you are still very active in your research. I see from different.
चल रहा थोड़ा बहुत कर रहे थे.
नहीं one has to be active. ठीक है.
सर इसीलिए कहते हैं ना कि जीवन चलने का नाम, बस रुकना नहीं है, जबरदस्ती. You have to keep pushing irrespective of age and all, because that is only the way we have to create positive aura around us. So keep working all the time, and that we have to learn from scientists we have to learn from scientists
थोड़ा सा
like
अपने लाइफस्टाइल को अपन चेंज कर लें.
Yes, sir. Yeah, right,
Thank you,
sir.
madam. Thank you so much. Sir.
Thank you,
Just,
sir.
just थोड़ा सा लाइफस्टाइल चेंज करके हम ज्यादा बेटर तरीके से रह सकते हैं. So
Yeah, sir.
it's a
Of course,
change in
yes, sir.
lifestyle.
Very true, sir. Yeah, thank you, sir.
Yeah, with some positive, with positive thoughts and like that. So everything is fine. Great, great. Nice to see you, everyone. Thank you.
Okay, thank you,
Namaskar.
thank you,
Thank you,
thank
sir.
you,
Thank you.
Namaskar.
Thank you, sir, for enlightening session. Now we know spices not only enhance the taste of food, but is good for heart as well. Now we'll take a 10-minute break and then we'll start with oral presentations. So hopefully you will all rejoin with us. Thank you.
Thank you, thank you. Thanks to everybody. Singh sir, namaskar. Sabko. Pattu sahab, Bhavi ji, sabko namaskar. Thank you.
Okay, namaskar, sir. Thank you so much, sir.
Okay.
Thank you.
Namaskar,
I have
namaskar.
to leave because I have to go somewhere. Thank you.
Okay,
But
okay, thank you.
we'll meet soon.
Okay,
Sure, sir.
thank you,
Sure.
sir. Thank you.
Inshallah.
Thank
Thank
you.
you, sir.
Thank you. Thank you. और बंद करता हूँ। हाँ, पापा। अरे मेरे बच्चों हमें बात नहीं करना।
Now we are going to have the chairperson of this session would be Subhash Chandra from Ajmer University. He will join us shortly. Till that, we will continue presentations. Our first participant would be Mani Patel from Department of Life Sciences, Gujarat University, Gujarat. Margi Patel.
Yes, present, ma'am.
Okay.
Shall I start?
Yes, you can start.
Okay, thank you. Good afternoon, all. My name is Margi Patel from Gujarat. First of all, thanks to University of Rajasthan and International Academy of Physical Sciences, Allahabad, for organizing such a wonderful conference. And most importantly, it is the free of cost. My topic is characterization of plant growth promoting traits of hollow-tolerant rhizobacteria. Next slide, please. So the climate change, a hot topic of current era, has affected world in different ways, and the rapid increase in saline landscape is one of them. Ultimately leads to global food insecurity and reduced agriculture productivity. The extent of salinity affected areas is expected to cover about 50% of total agriculture land by 2050, and as highlighted in global agriculture productivity index, the current growth rate of agriculture production is not enough to meet the projected food demand of 10 billion people in 2050. So both the scenario of increase in salinity and population ultimately leads to global food insecurity and reduced agriculture productivity. Next slide, please. High salinity significantly affects plant physiological aspect that resulting in the decrease in germination rates, fresh and dry biomass, photosynthetic pigments, essential nutrients uptake, and most importantly in the loss of final crop yields. To counteract the adverse effect of soil stress on plant growth, hollow-tolerant plant growth promoting rhizobacteria are one of promising candidates that can be used as an eco-friendly, low-cost technology to fight against abiotic stresses for sustainable production. The diverse group of bacteria in the close association with roots and capable of stimulating plant growth by any mechanism of action that are referred to as the plant growth promoting bacteria. Next slide, please. The objective of this study is to isolate and characterize the potent hollow-tolerant PGPR and to evaluate the effect on plant physiological and biochemical parameters. For that, PGPR strains were isolated from rhizosphere of hollow-tolerant plants Suida nudiflora from the saline desert of Kutch, Gujarat. Then isolates were screened for various plant growth promoting traits like phytohormone production, indole acetic acid, gibberellic acid, ammonia, hydrogen cyanide, ACC deaminase enzymes, CDRO4, hydrolytic enzymes, and also check the tolerance to different abiotic stresses like pH, temperature, drought, and salinity. Also check the potassium, zinc, and phosphate solubilization, nitrogen fixation, and also in vivo for the growth promotion of plant Amaranthus viridis in saline condition. Next slide, please. As you can see, the selected strain was able to produce indole acetic acid, ammonia, hydrogen cyanide, and able to fix the nitrogen. Pink color formation shows the production of indole acetic acid, and quantitatively it is the 35.7 microgram per ml. And the brown color development shows the ammonia production, and quantity is the 2.4 micromole per ml. Then hydrogen cyanide production in the conformation by the change of Wortmann filter paper number 1 from yellow to brown, and the plate yellow to blue is the conformation of nitrogen fixation ability of the rhizobacteria. This strain also able to solubilize the phosphate, potassium and zinc, and able to produce CDRO4 and some hydrolytic enzymes like amylase, chitinase and cellulase. It was able to tolerate abiotic stresses like salt, temperature, drought and pH. It was not able to grow at the 30% PEG in the drought tolerance. Otherwise, it was able to grow at 50% salt, 45 per degree Celsius and 9 pH. Next slide, please. This image shows the Amaranthus viridis plants, treated and non-treated plants. You can easily see that the highest germination percentage and also the overall plant growth by shoot and root height, fresh and dry weight. The treated with rhizobacteria plants shows the maximum properties of the plant growth promotion. And the selected strain was not able to produce bralic acid, ACC aminase enzyme, and the hydrolytic enzyme pectinase and protease. All the port experiments were carried out in randomized block design, including 4 treatments in each normal and saline. So saline condition is provided with the addition of NaCl and the saline condition at the level of electric conductivity 4 decisiemens per meter. Next slide, please. You can see that rhizobacteria was able to increase the plant chlorophyll content, chlorine content, glycine betaine, total soluble sugar, and the total free amino acid content then as compared to the non-inoculated plants. And the hydrogen cyanide, hydrogen peroxide, it is the highly toxic for plants, and the rhizobacteria was able to decrease this H2O2 content in the treated plants. Next slide, please. Then the defensive antioxidant enzymes like ascorbate peroxidase, superoxide dismutase, glutathione reductase, and catalase was increased in the rhizobacteria treated plants, and also the macro and micronutrients was increased in the PGPR treated plants. Next slide, please. The current findings suggest that halotolerant Bacillus methylotropicus has great potential to improve Amaranthus viridis productivity and tolerance under salinity by eliminating the harmful effects of salt stress on plant growth. This strain would be a promising candidate as biofertilizers as an alternative to chemical fertilizer under salt stress conditions, and eco-friendly strategy to increase salt stress and crop yield. Future studies are needed to investigate the mechanisms of induced salinity tolerance by identified PGPR strain and molecular level. Next slide, please. Thank you so much, everyone.
I would like to invite our next speaker, Shweta Singh from IIS University, Jaipur.
Am I audible?
Yes, ma'am, you're audible.
I'm Shweta Singh from the IIS University, and my topic is sustainable utilization of leaf litter through composting using natural and commercial activators. Next. Environmental degradation is a major threat confronting the world, and the primary causes of environmental degradation are rapid growth of population, overutilization of natural resources, establishment of different multinational companies and local industries, air pollution, water pollution, deforestation, thinning of the ozone layer, global warming, sanitation and outbreak of diseases are the problems that are endangering human life. And besides these, the problem of waste disposal cannot be ruled out. Solid waste is of next slide, please. Solid waste is often referred to as third pollution after air and water, arises from the different activities of humans. Solid waste can be either organic or inorganic, such as product packaging, grass clipping, leaf litter, furniture, clothing, kitchen refuse, paper, etc., produced in our society. Approximately 80 to 90% of municipal solid waste is disposed of in landfills without appropriate management procedures or open dumping, resulting in contamination of the air, water, and soil. According to the data obtained from Ministry of Housing and Urban Affairs, Swachhata Sandesh newsletter, 1,47,613 metric tons of solid garbage is created per day from 84,475 wards as of January 2020. The Indian government also announced the Swachh Bharat mission on October 2nd, 2014 for a 5-year period with the goal of producing a clean India. The solid waste municipal rules were also updated and notified by the Ministry of Environment, Forest and Climate Change in April 2016. Next slide, please. There are various sources of solid waste, such as agriculture waste, commercial waste, e-waste, construction waste, biomedical waste, municipal solid waste. Next slide, please. Leaf litter accumulation in urban and suburban locations, such as sidewalks, lawns, and playgrounds, adds to the overall problem of municipal solid waste disposal. In many countries, including India, leaf litter is often piled up and set on fire. The burning of leaf litter adds to the air pollution and causes several health problems. Leaf litter accumulated in forests can also cause serious fire incidents. Leaf litter is a very potential energy resource if managed properly and biologically converted to organic manure. Next slide, please. Various sources of leaf litter are playgrounds, roads, rail lines, lawns, forests, sidewalks. Next slide, please. Compost is becoming an important aspect in the quest to increase productivity of crops in an environment-friendly way. Composting is a biological process to convert biodegradable waste into nutrient-rich compost. Leaf litter has a complex structure that cannot degrade easily. So to accelerate the process, the decomposition process of leaf litter during composting, different type of activators are used. These activators can either be natural or commercial. Next slide, please. Natural activators which we can use during the composting process can be cow dung, cow urine, jaggery powder, sugarcane juice, lime powder, and commercial activators are enzyme powder, bacterial and fungal inoculums, composting culture powder, liquid decomposer. Next slide, please. The study will propose an alternative approach to waste management, since the waste will neither be landfilled nor burned, would be used as a resource by the process of composting. Leaf litter management through composting, if applied on large scale, it will help to reduce air pollution produced by leaf litter burning. Sustainable utilization and management of leaf litter through composting can be successfully applied in home gardening and agriculture purposes. Different types of nutrient-rich compost prepared can be further used for organic farming to grow vegetables, fruits, cereals, etc. Leaf litter utilization and management will help in making the residential areas cleaner, and people will get to live in a sustainable environment. Thank you.
ऐसा है ज्यादा टाइम लगेगा लगता है तो फोन टू साइड से स्किप कर दे क्लास वाले को तो क्या मैं करते हैं तो प्रेजेंटेशन करें डेफिनेटली
Now I would like to call upon our next speaker, Jyoti Sharma from IAS University, Jaipur.
Am I audible, ma'am?
Yes, ma'am, you are audible.
Ma'am, screen share.
Is the screen visible to you?
Yes, ma'am. Ma'am, please.
Is this your PPT, Jyoti
Yes.
Sharma?
Yes, ma'am.
You can go ahead with your presentation.
Okay, ma'am. Good afternoon, everyone. My name is Jyoti Sharma. I'm research scholar from IIS University, Jaipur. My topic is today. I'm going to presentation. My topic is comparative study of liquid biofertilizer and carrier-based biofertilizer on various growth and biochemical parameters of vegetable pea. Next slide, please. India is the second largest populous country in the world, mostly depend on agriculture for living. Agriculture occupies a very important place in the economic of India. It's backbone of our ecosystem. In this era, modern agriculture, some techniques are used for farming. Chemical fertilizer is one of them, but these are not eco-friendly. Next. Some harmful effects of chemical fertilizer, that is reduced legume root nodulations, nutrient imbalance in plants, decreases in soil life, soil humus reduction, and environmental hazards such as water pollution, soil pollution, etc. Next slide. Due to this limitation of chemical fertilizer, the use of biofertilizer increases day by day. Biofertilizer are defined as biological active products or microbial inoculants of bacteria, algae, and fungi, which may help biological nitrogen fixation for the benefits of plants. There are four formulation of biofertilizers: liquid biofertilizers, carrier-based biofertilizers, granular biofertilizer, and encapsulated biofertilizer. Out of these four formulations, the liquid and carrier-based biofertilizer are being used for the present study. Next, please. Next slide. Liquid biofertilizers are microbial preparation containing specific beneficial microorganisms which are capable of fixing or solubilizing plant nutrients by their biological activity. These are increasingly available in the market as one of the alternative to chemical fertilizer and pesticides. These are effective for crop plants. Carrier-based biofertilizers are the biofertilizer prepared with the help of activated charcoal, which act as a audible, ma'am?
Please go ahead. You are audible, ma'am. Please go ahead with your presentation.
Ma'am, next slide.
Sir, next slide.
Comparison of liquid and carrier-based biofertilizers. Liquid biofertilizers are tolerant to high temperature, but carrier-based are so sensitive. Moisture retention capacity of liquid biofertilizer is high and carrier is low. Shelf life of liquid biofertilizer are longer, 12 to 24 months, and carrier-based are so short, 3 to 4 months. Transportation cost of liquid biofertilizer are low and carrier-based are high. Considering this, it is determined that liquid biofertilizer are best alternative for the conventional carrier-based biofertilizer in the modern agriculture, which help in the sustainable global food production. Next, ma'am. Next slide. We have selected vegetable pea as a test species. The scientific classification of vegetable pea: kingdom Plantae, order Fabaceae, family Fabaceae, genus Pisum, species Sativum. Pea is used as fresh or processed vegetable. In India, pea is extensively cultivated in UP, Bihar, and Madhya Pradesh. Pea is an annual plant with a life cycle of 1 year. It is a cool season crop. Next slide. The following treatment combinations, the following 3 combination of treatment with 3 replication of each treatment were maintained for the present study. T1 is carrier-based biofertilizer, T2 is liquid-based biofertilizer, and T3 is absolute control. Next, ma'am. Results, plant sampling and analysis. For the purpose of analysis, the experimental plant samples were collected at the time of pre, peak, and post flowering stage of the test species and were analyzed for the following parameters: physical parameters such as root length, shoot length, and total length. And second is root dry weight, shoot dry weight, and total dry weight. And third is plant phosphorus biochemical. Next. The result of my study, physical parameters of vegetable pea, pre, peak, and post flowering stage. We observe T2 treatment. We have observed the highest result, that is total length of plant, root and shoot, total dry weight, root and shoot, and shoot dry weight, root dry weight, and total dry weight. And lowest result in T3, absolute control. And plant phosphorus at pre-peak and post-flowering stage, we have observed T2 treatment in highest plant phosphorus and lowest in T3 treatment. Next. Thank you. Thanks.
Now I would like to call upon our next presenter, Tanisha Ameria from Department of Environmental Sciences, IIS University, Jaipur. Tanisha. Are you able to see your slide, Tanisha? Are the slides visible to you, Tanisha?
Yes.
You can go ahead with your presentation.
Good afternoon, all. Good afternoon, all. The topic of my study is spatiotemporal assessment of LULC and heat islands of Jaipur district. And the time frame that was taken by me was from 2009 to 2019. Please change. The term land use and land cover, these two terms are often used interchangeably, but they differ in their two senses. That is, the term land cover refers to the natural setting of the environment or on the earth surface. All the natural features that are present on the earth surface is referred to land cover, whereas the land use includes how the humans use this land for the social, economic, cultural, political, or developmental activities. And this land use in turn influences the land cover and the environment in total. And one such impact is the increase in temperature and the formation of urban heat island. Please next. Now the methodology. Next. The study area chosen for the study was Jaipur district, that is present in the Rajasthan state. Next. Satellite data. Next. The satellite data taken for the study was Landsat 7 for year 2009 and for Landsat 8 for 2019. For the image pre-processing, the band 1, 2, and 3 of Landsat 7 were taken for making the composite, and band 2, 3, and 4 of Landsat 8 were taken for making the composite image for the year 2019. And then using the shape file, the composed images were clipped. Next. The estimation of land use land cover. For estimating the land use land cover, first the satellite images of both the years were taken. After pre-processing, the LVLC on the images, the supervised classification method was taken, and from supervised classification method, maximum likelihood classification algorithm was been applied, and then the LVLC, the land use land cover maps were created by categorizing the area into 3 classes. That is, first the built-up area, which includes residential, commercial, transportation, etc. The second class was vegetation, which included forest, plantation, agriculture, all the green cover of the city or the district, and rest were taken in the category that is other. And then after making the maps for 2009 and 19, the change detection was being assessed. Next. And the software that was utilized for the entire study was ArcGIS Pro. These were the LULC maps that were being created using the ArcGIS Pro and using the algorithm. And you can it's clearly visible that during the decade, the built-up area has increased a lot. And please change. Apart from the built-up area, we can see that the built-up area has almost increased to 27%, and with this simultaneously vegetation has also been observed to be increased, but the influence of this increase in vegetation in contrast to built-up area was very negligible. So it was not having that much impact, such that the impact that was having the next. Then for classifying the wait. Please back. Okay, just. For estimating the assessing the relationship between land use land cover and urban heat island, first the LST maps were being created, and for creating the LST maps, first the Landsat 8 thermal band 10 and Landsat 7 thermal band 6 were taken for both the years. From these images, digital number values were being converted into spectral radiance value using the formula, and all the values that were given in the table is available in the metadata file of these satellites. Next. And then after converting the digital number into spectral radiance, these spectral radiance values were being converted into brightness temperatures. Firstly, these brightness temperatures were in Kelvin. Next, then it were being converted into the Celsius. Next, using this formula, and after converting the brightness temperatures into degrees Celsius, simultaneously normalized difference vegetation index was being calculated, propagation vegetation was calculated, emissivity was calculated, and finally the LST values were being assessed or measured by putting all these values in this formula. And after estimating the land surface temperatures, the land surface temperature maps were being reclassified into different categories of surface temperatures. And on this reclassified map, the urban vector file of Jaipur district was overimposed, and then urban heat islands were identified, and then wind was correlated with the LULC maps. Next. And for identifying the urban heat island, the criteria that was used was the one which was given on the basis of heat wave categories that was given by Indian Meteorological Department. And the reason behind using this criteria was first that these temperature categories that were given by the Indian Meteorological Department were found to be very dreadful for human health and environment according to the literatures, and no research has been undertaken on classifying the urban heat islands on these heat wave categories as still our knowledge. So this makes the study novel. And according to this IMD categories, when the actual maximum temperature was found to be greater than or equal to be 45 degrees Celsius, then it is a condition of heat wave. So we took this heat wave condition as a urban heat island category. Then when the actual maximum temperature was found to be greater than or equal to 47 degrees Celsius, then there is a condition of severe heat wave. And this condition we took as a severe heat wave category, severe heat urban heat island category. Next. And so this is the result. In 2009, the temperature was in the overall decade, we found that almost there is a 5% increase in the temperature, 5 degrees Celsius increase in the temperature. And in 2009, only urban heat islands were found that was having the temperature greater than up to 45 degrees Celsius. But there were no severe urban heat islands were being observed in this year. Next. But in the year 2019, the temperature was found to be, the land surface temperature was found to be intensified. Furthermore, severe urban heat islands were also found. And majorly they were found in the region of Jaipur city. So with this, the study concluded that the district has undergone a rapid urbanization and industrialization during the decade, and there is a dire need for continuous monitoring of land use, land cover, along with the development of rational, scientific, and sustainable urban land use plan. The study also highlights the key urban areas for work, that is majorly, as we have seen in the maps, through the maps of LULC, that the urban area has increased in southwest and majorly in the southwest and northwest region. So there is a need for working in those areas. Also, it represents the potential use of study to understand the driving forces, the key hotspots, and to develop sustainable land use plans for such areas and policies for reducing and maintaining the balance in ecosystem. Thank you.
Now I would like to invite our next speaker, Dipan Sharma from Tripura University.
Ma'am, am I audible?
Yes, sir, you're audible.
Okay, ma'am, please share the screen.
Sir, are the slides visible to you?
Yes, ma'am, yes. May I start, ma'am? May I start?
Yes, sir, you can go ahead with your presentation.
Okay, ma'am. Okay, thank you. Myself, Dipan Sharma, presenting my topic title is Green Synthesis of Silver Nanoparticles using Alphania galanga, L.wilt leaf extract and its antibacterial activity. Next slide, please. Introduction. Nanotechnology is one of the most active and promising technologies applied in all areas of science. Over the past decades, research on nanoparticles has been developing rapidly due to its unique properties and application in various fields. Thus, they are getting popular as natural alternatives to synthetic chemicals. Next slide, please. Biosynthesis employs biological means such as bacteria, fungi, and plant extracts to synthesize nanoparticles. Among them, plant-based nanoparticle synthesis can be simple and beneficial above other biological agents, as it reduces the complicated process of maintaining fungi or bacterial cell cultures. For the production of the best capping materials for stabilization, silver nanoparticles synthesized from plant extracts are used. Plant extracts show potential benefit over microorganisms, as reported by various researchers. So currently, silver nanoparticles are the most widely commercialized nanoparticles that are used as antimicrobials in various consumer products ranging from cosmetics, detergents, water purification systems, etc. Next slide, please. The present study was undertaken with the following objectives: green synthesis of silver nanoparticles using leaves of Alphania galanga, characterization of biosynthetic silver nanoparticles, investigate the antibacterial activity of synthesized silver nanoparticles. Next slide, please. Methodology. The Alphania galanga leaves are collected and washing several times by distilled water, and then about 10 grams finely cut leaves are kept in a beaker containing 100 ml of distilled water and boiled for 30 minutes. Then the extract was cooled down and filtered through Whatman filter for number 1. For green synthesis silver nanoparticle, 1 millimolar of silver nitrate solution prepared and then 10 ml of filtered. Each plant extract was added to 90 ml of silver nitrate solution and keeping this concentration at 1 millimolar. For characterization of biosynthetic silver nanoparticles, UV visual spectroscopy, AFM and SEM and EDX was carried out. Next slide, please. As a result, a distinct color change was observed after addition of aqueous extract to silver nitrate solution. The color of the solution changed from transparent to reddish brown, indicating the formation of silver nanoparticles. The figure showing Alphina galanga plant, the plant leaf extract, and C showing the initial mixture of silver nanoparticle and plant extract mixture, and the final result synthesis silver nanoparticles after 48 hours. Next slide, please. UVB spectroscopy analysis performed continuous scanning from 350 to 800 nanometer, and the nanoparticle solution showed maximum absorbance at 428 nanometer after 72 hours. Next slide, please. FTH spectral analysis performed by contrast scanning. FTH carried out to identify the functional group present within the silver nanoparticles. The results showed the presence of hydroxyl group, nitrate group, aromatic alkaline double bond, carbon double bond group, etc. Next slide, please. Please, next slide. The AFM analysis, ma'am, please, previous slide. AFM analysis in the nanoparticle is showing the maximum particles at 14 nanometer and after surface. Next slide, please. Next slide. Same image show the nanoparticle shape like rocky spherical leaves and few irregular granular complex fused agglomerates. Agglomerates ranging from 35 nanometer to 50 nanometer. Next slide, please. Energy dispersive X-ray analysis show the silver which comes the synthesis of silver in high quantity in negligible traces of nitrogen, carbon, oxygen as impurity. It also noted the presence of aluminum because of sample preparation upon aluminum foil. Next slide, please. The antibacterial activity was tested against 4 bacterial, strains: Bacillus subtilis, Staphylococcus aureus, E. coli, and Pseudomonas aeruginosa. The antibacterial activity highest present in 100 microgram against the bacteria Pseudomonas aeruginosa followed by Bacillus subtilis. Next slide, please. As concerned, the green approach is an easy, extremely low energy based, eco-friendly. It does not require the use of any kind of hazardous chemicals, reagents for reduction and processing. Elemental analysis of nanoparticles confirms the presence of elemental silver with negligible trace of carbon, oxygen, nitrogen, fluorine as an impurity. The synthetic silver nanoparticles have shown antibacterial activity against gram-positive and gram-negative bacteria, pathogenic bacteria, which could establish their application in biomedicine. Thank you, ma'am. Ma'am, next slide. Thank you. हमारा था कि बने हमारा स्क्रीन था कि बने okay.
Now I would like to call upon our next speaker, Dr. Nazia. Dr. Nazia, Head of the Department Chemistry, Mustaji Maharaj University. Dr. Nazia.
Yes, ma'am. Am I audible?
Yes, ma'am, you're audible. You can start with your presentation.
Yes. Are you sharing, ma'am?
Yes, ma'am. Are the slides visible?
Okay, thank you. Yes, ma'am. Thanks. May I start?
Yes, ma'am. Yes, ma'am. You may go
Okay,
ahead.
thank you. Okay, thank you. A very good afternoon to respected chairman and all the present over here. I, Dr. Nazia Akhtar Rashidi. First of all, I am very much thankful for giving me this opportunity of presentation of my research paper on the topic
plastic pollution and plastic recycling. Friends, no one can deny to the fact that plastic is an incredibly useful material, and every day we make use of plastic. In fact, human has been making use of plastic since from beginning. Since from 1907, the first plastic that was developed was the bakelite, and since then we are making use of plastic, and it has become very common to us. Even though we have a very big question in front of us: is plastic good or bad? Plastic strength as a material is very bad for the environment because plastic is non-biodegradable and it poses a serious damage to our environment. It pollutes earth, air, and water. When we dump it in the air, when we dump it in the land, it infertilizes the land. When we burn the plastic, it releases the harmful gases, greenhouse gases such as CO2, carbon dioxide, Cl2, etc., and it pollutes our atmosphere. When it throw in the sea, then it kills the marine life and other habitants. In fact, it affects all the species which are in the food chain. So see how it is dangerous. Next one, next slide. So today, plastic pollution is one of the major environmental pollution, and plastic causes serious damage to our environment, both during its production as well as disposal. Next one. Now let me move to the solution over it. Plastic waste management is the only solution to get rid of this serious problem. There are the different stages when we go to the plastic waste management. The first one is reduce, means to avoid the production of plastic waste. Next is the reuse, means we have to make use of plastic as many times as possible. Next is the recycle. After the reuse of the plastic, we should go to the recycling. Next one is the recovery, means we have to turn the plastic waste into the useful one, that is energy from the waste. And the last one is the dispose it in a responsible way. So we can see that out of all these options, the first option is the most favorable option because it reduces the plastic, and the last option, that is the disposal of the plastic, is the least favorable option. So when we go to the plastic recycling, it is one of the important option again. Next one. Next slide, please. Plastic recycling. Plastic recycling is beneficial in many ways. Again, it involves the different stages. This flowcharts show the different stages involved in the plastic recycling. So first one is the collection and next is the cleaning. So very first one, that is when we go to the plastic recycling, we have to collect the plastic waste from the different fields: industrial waste, commercial waste, agriculture, and municipal waste. After collection, we have to clean it up to make sure that all the contaminants have been eliminated from the plastic waste. And then next stage is the sorting. Remember friends, plastic recycling is a crucial matter. So to recycle or not to cycle is again a big question in front of us. So for that purpose, sorting is very, very essential. So when we go to the sorting, we have to identify the plastic on the basis of their quality as well as grade. This is very important step. After sorting, we have to go for the size reduction. It is also called as the tearing of the plastic. Here, plastic is turned into the small pieces and granules, and then it is subjected for the melting. And the last step is the extrusion. Extrusion is one of the important steps in plastic recycling. In this step, the plastic materials paste, which is converted into the small granules, it is subjected to the extrusion machine. It is allowed to flow from it, and then it is taken out from the opening. The method is very exactly similar to the taking out of the toothpaste from the toothpaste tube. So here, plastic waste is converted into the wonderful plastic material which we make use in our daily life. Next one, next slide. So in short, we can say that there are the 5 main stages of plastic recycling. It starts from the plastic identification, where we have to identify the plastic on the basis of its quality and grades. Next is the washing of the impurity to eliminate the contamination. Next is the tearing of the plastic to convert it into small granules or pieces. Next, again, we have to re-identify the quality of the plastic, and last step is the extrusion of the plastic. Next one. So as a conclusion part, summing it up as a conclusion part, recycling plastic is beneficial in many ways. The landfill that pollutes the environment with the tons of unused plastic will start emptying, and moreover, this as plastic is derived from the oil, which is a non-renewable natural resources, so it will solve the energy problem also. And by the recycling and reuse of the plastic, we can contribute in reducing the global warming too. So, so when we go to the recycling of the plastic, it is the I think it is a most important step for saving our environment. Thank you. Next one. Thanks a lot.
Now I would like to invite our next speaker, Ms. Sushila from Department of Botany, University of Rajasthan. Sushila ma'am, are you there?
Yes, ma'am.
Are the slides visible to you, ma'am?
Yes, ma'am. Yes.
Okay, ma'am, you can go ahead with your presentation.
Just one minute. Okay, ma'am. Thank you. Myself, Susila from Botany Department, University of Rajasthan, Jaipur. My topic is floristic analysis of wild edible fruits in Sikar district of Rajasthan. Next slide, please. Wild edible fruits are available in their natural habitat. These have better nutritional constituents like protein, minerals, etc. in comparison to cultivars. There are some interesting wild edible fruits observed during field study. My field study is Sikar district of Rajasthan. The study area has arid climatic conditions followed by semi-arid conditions. Some plants which are available in Sikar district for wild edible fruits. Prosopis cineraria, family Fabaceae, Momordica dioica, family Cucurbitaceae, Cucumis milo, variety Agrestis, Gigipus pneumolaria, Bohinia racemosa, variety Fabaceae, Caperis decidua, Citrullus colosynthesis, Leptadenia pyrotechnica, Ciarcia mysorensis, Cordia garaf, family Boraginaceae are observed during exploration. Among these plants, herbs and shrubs are equally in number, followed by tree species. Next slide, please. The term wild edible fruits represented as any part of the reproductive structure of angiosperms and consider any undomesticated products extract from wild or managed landscape as wild. Sourcing food from the wild has been closely entangled with humanity for millions of years. A diverse range of wild cultivated plants and their parts, example, leafy shoots, seeds, underground organs, and flowers are still being consumed regularly and complement human adaptability and a very variety of human gastronomic choice. They tend to supplement proteins, essential minerals, micronutrients, and vitamins that enrich dietary quality and thus provide an affordable source of nutrition for rural and semi-urban societies across culture and continents. Next slide, please. Next slide, please. Study area. Collection and identification of wild edible fruits from Sikar district to explore wild edible fruits plant potential in Sikar district. Duration of survey, field visit, herbarium seed preparation. Duration of survey, preparation a list of plant species that grow in different seasons, like rainy season, winter season, etc. Duration of the study work is 2 year maximum. Field study, field visit, frequently field visit the study area and collection of different plant species were done. Herbarium seed preparation, herbarium seeds of the collected plant will be prepared, documented and submitted to the RUBL. Next slide, please. Result and discussion. To study show present status of wild edible fruits of the study area. The main wild edible fruits are Ciarcia myosorensis, commonly known as dancere, Momodica dioica cacoda, Cucumis milo variety agrestis kachri, Leptadenia pyrotechnica, commonly known as keep, are noticed from the field visit of Sikar district. Next slide, please. First diagram shows Ciarcia myosorensis, synonym Rus myosorensis dancere. Second slide is Cucumis milo variety agrestis. And third diagram show Leptadenia pyrotechnica. Next Momodica dioica. Next slide, please. Thank you, ma'am.
मैडम बिल्कुल अगर आपने खाई तो मैं खाना भी नहीं खा सकती निधि राजौरिया
Yes, ma'am. I'm audible.
Yes, ma'am, you're audible. You can start with your presentation.
Okay. Please screen share. Hello. Good afternoon, everyone. Myself, Nidhi Rajoria. I'm going to present my presentation. My topic is occupational exposure to air pollutants in an underground parking. Next slide. Indoor air quality plays a significant role in determining the health status of an individual. Occupational exposure to indoor air pollutants at workplace is a major problem because the worker spend most of time, like 80 to 90% in the indoor environment, thus are exposure to toxic waste air in this environment, like as hospitals, restaurants, garages, indoor parking, temples, beauty salons, offices, and enclosed buildings, etc. The level of exposure of air pollutions at the workplace can be much higher than the outdoor. According to this scientific Rosenfeld et al. 1995, Chen and Can 2008, and Zhou et al. 2011. Next slide, please. What is an indoor air? Airborne particles, this is like diesel, exhaust, carbon black, dust, smoke, fibers, plant matter, hair pollen, etc. Household odor and gases, cooking odors, fat, smells, cigarettes, smoke, chemicals, skin or drain smells. And third one is volatile organic compounds, paints, clothes, and varnishes, wood, preservatives, cleaning supplies, offices, equipment, and furnitures. And last is microorganisms, bacteria, mold, yeast, mites, and virus. Next slide. Major indoor air pollutants are such as many pollutants, but here is major indoor air pollutants. First is particulate matter, PM. Second is SO2, and third NO2, nitrogen dioxide, and fourth is volatile organic compound, and fifth polycyclic aromatic hydrocarbon, PHS. Next slide. Major health effects of air pollutions, pollutants PM, and sources: cleaning, working, cooking, dust, soot, smoke, diesel engines, vehicle exhaust, wood stoves, etc. And health effects is reduced lung function, respiratory symptoms, cancer, cardiopulmonary, mortality and heart rate variability, blood viscosity, destabilization, etc. Second is VOC, volatile organic compound. Sources is in indoor, beauty products, fuel combustions, paints, automotive products, welding, furnishing, etc. Health effects: respiratory disease, hematological problems, cancer, skin irritation, fatigue, headache, dizziness, weakness, confusion, insomnia, and nausea, etc. Next slide, please. PHS sources is indoor, motor vehicle exhaust, coal, fuel, stores, smoking, cooking, domestic heating, and fireplaces, sanitizers, etc. And health effects is eye irritations, nose and throat, nausea, vomiting, diarrhea, breakdown, red blood cells, asthma, lung functions, cataracts, decreased immune function, confusions, cause headache and dizziness, etc. Next is SO2, sulfur dioxide. Sulfur dioxide sources is indoor, mostly carbon fossil fuel combustion, diesel, unvented kerosene, vehicle exhaust, space heaters and domestic coal burning, and etc. And health effect is asthma, premature mortality, sensory functions, irritation, nose, throat, eye, breathing difficulties, lung function reduced, loss of vision, etc. And last is NO2, gas fired. Sources is gas fired, appliance, wood, stoves, oven, unvented gas, space heaters, unvented kerosene, tobacco smoke, burning of fuels, emissions from underground parking, etc. And health effects is respiratory infections, loss of appetite, corroded teeth, coughing, wheezing, breathing problems, asthma, and etc. Next slide, please. 7 million people die every year from air pollutions. Effect of air pollution on health. First is hair damage, eye and mucous membrane infection, respiratory disease, liver, spleen and blood damage, and bone damage, fatigue and headache, noxicity, nervous system damage, cardiovascular damage, skin damage, intestinal damage, and reproductive system damage. Next slide, please. Next. Thank you. Thank you so much.
It is a great honor for me to introduce Dr. Subhash Chandra Sir, Professor and Head Department of Zoology, Maharishi Dayanand Saraswati University, Ajmer. Sir has an experience in teaching and research of more than 20 years. His main area of research is biodiversity conservation, natural resource conservation, biodegradation, and animal behavior. He has published around 40 research papers and chapters in the journals of national and international repute. We welcome you, sir. Sir has joined a bit late. We welcome you, sir. Am I audible to you, sir? Sir, am I audible to you? Subhash Chandra, sir. Sir, am I audible to you? I think there's some technical issue So sir will join us after a while. We'll begin with our next presenter, Suman Paul from Department of Botany, Tripura University. Suman Paul.
Am I audible, ma'am?
Yes, sir, you are audible.
Okay. Kindly share the screen. Okay. May I continue?
Yes, sir. Yes, sir, you can begin your
Okay.
presentation.
Good afternoon, everyone. Myself, Shumon Pal from Department of Botany, Tripura University. I will be presenting my research work entitled Impact of Land Use Change and Agricultural Intensification on Plant Pollinator Diversity of Tripura, Northeastern India. Next slide, please. As all of us aware of the fact that pollination is an essential ecosystem service and majority of the flowering plants depends on pollination for the successful reproduction and their survival. It has been estimated that approximately 78 to 94% of the flowering plants depends on pollination. Additionally, 75% of the crop plants that we cultivate depends on pollination. In other words, we can say that the every one of the 3 bites of food that we eat depends on pollination. More recently, urbanization and land use change has been reported to affect pollinator diversity and plant pollinator networks by changing the resource availability to the pollinators. Next slide, please. Keeping this background on mind, the present study was focused to determine the pollinator diversity in 3 different land use systems. Additionally, we tried to determine the pollinator abundance and richness in 3 different land use systems. Next slide, please. To do so, we have selected 3 land use systems. Accordingly, agricultural lands, home gardens, and semi-natural habitat sites. Pollinator sampling and identification, pollinator sampling and data collection was done using transect work method and focal observation. We have collected the insect from the field and identified with consultation with expert taxonomists from different institutes. Then we try to assess the diversity of pollinators using different diversity indices. Next slide, please. We have observed in total 74 morphospecies of pollinators represented in 18 families and 4 orders. Most dominant order was Lepidoptera and Hymenoptera, represented by 45% and 33%, whereas Diptera and Coleoptera was less abundant, represented with only 15 and 7% respectively. All the pollinators are found to visit 24 flowering plants comprising of 10 families that were present in the 3 different sites. Next slide, please. The figure 1 represents the pollinator abundance and richness in 3 different land use systems. As we can see in the figure, that pollinator abundance and richness both were highest in the agricultural farm compared to the other 2 land use systems. Next slide, please. Here we can see that pollinator diversity in 3 different systems, 3 different land use system, and the diversity index Shannon and Simpson index suggests that semi-natural vegetation sites harbor rich diversity of pollinators compared to the agricultural farms and home gardens. Next slide, please. Our present study revealed that pollinator abundance and richness was higher in the agricultural land, and the diversity of pollinator was found to be higher in the natural habitat because the agricultural land harbors rich floral resource diversity compared to the semi-natural sites. Additionally, we found that butterfly species were preferably visiting the semi-natural sites, and bees and fly species were visiting home garden and agricultural sites more than the semi-natural sites. These type of studies may help the farmland owners as well as the environmental policymakers to design environmental friendly or pollinator friendly ecosystem for the future. Next slide, please. Lastly, I would like to thank you all for listening.
Subhash Chandra sir, am I audible to you? Subhash Chandra sir, are you getting my voice, sir? So we are not able to hear you. So you're not audible. So we are not getting your voice. I think there's a technical issue over here. So meanwhile, we'll move on to our next presenter, Dr. Manvi Malwal from Botany Department, Chandigarh. Botany Department, Government College for Girls, Chandigarh. Dr. Manvi.
Good afternoon, all of you. Am I audible?
Yes, ma'am, you're audible.
Okay. My topic is novel Indian therapeutic plants against viral infection, and it is a review paper on the drug development perspective of herbal medicines. Next slide, please. Herb is originated from the Latin word herba. Herbal plants are utilized as food additive, folk medicines, and few spiritual activities like we worship banyan tea and tulsi commonly. Herb plants are systematically used in traditional system of medicine such as Unani, Ayurveda, and Chinese. About 8,000 herbal remedies have been codified in Ayush of India. World Health Organization has estimated that about 80% of the world population rely on herbal plants derived medicines from their primary health care. Most of us goes to the herbal plants in this COVID period also. They are considered to be safe for with minimal side effects as compared to the allopathic medicines. Next slide, please. These are the commonly herbal plants which we utilize in our kitchen also. Osimum we use in tea and there is ginger, turmeric and all these plants we mostly use in every remedies because it has potential activities like antimicrobial, antifungal, and a number of metabolites have been found all these plants like flavonoids, phytosterols. All these compounds have a number of activities like anti-cancerous and other biochemical activities. Next slide, please. Next slide, please. These herbal plants have been reported for number of activities, also like immunomodulatory activities. In SARS, there is these plants are mostly utilized as immunoboosters. In Ayush, they have codified 37 plants for immunoboosters, and many plants are utilized for antimicrobial activity due to their flavonoids and alkaloid compounds. Moraya, which is Kadi Patta, is also an important anti-cancerous plant which mostly we utilize in our cuisines. And a number of plants have hypolipidemic and anti-ulcer activities also. Next slide, please. The word coronavirus, the unexpected pandemic is an unprecedented challenge for public worldwide during these days, and it is the third zoonotic coronavirus breakout that has human-to-human transmission and raising global concern. And Ministry of Ayush issued a statement that Ayurveda immunosuppressing measure for self-care, which include the recommendation to take Chamarbrash formula. It contains around 87 plants and there are number of phytochemicals which are utilized. The utilization of herbal-based medicine to treat COVID-19 is similar to their use in treatment for severe acute respiratory syndrome disease. Plant species in SARS treatment include Lycoris, Artemisia, Pyrosia, Lindalia, Angelica, Panax, and Scutellaria rectia. All these plants I have studied from this Scopus website. A number of papers has been published and a number of phytochemistries have been identified which are utilized for SARS SARS activities. And the screening potential of bioactive principle from traditional herbal medicine is a viable status to be considered. Next slide, please. Herbal plants are naturally rich of active principle. They are used as primary health care and they are considered to be safe, cheap, and eco-friendly. And the blind dependence on synthetic drug is over, and the people are returning back to the natural with hope of safety and security. Thank you. Next slide. Thank you.
There's a request to all the participants. Please kindly stick to your time limit. Try to complete your presentation within 5 minutes. Next, I would like to invite our next speaker, Ms. Neha from Department of Biotechnology, Dwarka Das Govardhan Das Vaishnav College, Chennai, Tamil Nadu. Sneha from Department of Biotechnology, Dwarka Das Govardhan Das Vaishnav College, Chennai, Tamil Nadu. We'll move on to our next presenter for today, Aribam Devidas Sharma, Department of Economics, Manipur College, Imphal. Aribam Devadas Sharma. Aribam Devadas Sharma from Department of Economics.
Yes. Respected chairperson of this session, members of organizing committee, and everyone present here. Now today, I am from Manipur. Today I am presenting on biodiversity, conservation, and socio-economic issues confronting Loktak like a reality set. Next slide, please. Loktak Lake, located in Bisempur district of Manipur, is the largest freshwater wetland in Northeast India, in the whole Northeast India. It is also called only floating lake in the world and is regarded as the lifeline of the people of Manipur and soon will be on the verge of extinction. And this Loktak Lake is mainly become to the limelight of limelight because of the this Loktak Lake is also the only floating lake in the world. The Kabilamjau National Park, covering an area of 40 square kilometers, located in the southern zone, is the world's only floating national park. The last national habitat of highly endangered Manipur Bro Elter Dia Receivers LD local known as Sangai. This Sangai is an endemic and endangered species of Ildeers found only in Manipur. It is also the state animal of Manipur, and it is also in the verge of extinction. Next slide, please. It is why it is called Loktak floating lake. Why the Loktak floats? That is the one interesting part of this lake. It is called floating lake only because of the this Pumdi. The Pumdi is a
a local term we use here in Manipur. It is a porous and spongy biomass capable of floating due to buoyancy. The Phumdi house a large number of aquatic, semi-aquatic, and terrestrial flora and fauna. But the one interesting part here is that this Phumdi, why this Phumdi floats on the Loktak Lake? There is this interesting part. Why it floats on the lake? Because it is a unique phenomenon associated with the Phumdi, which makes the Sangai habitat unparalleled in the world. Unparalleled in the world. Mainly because this Phumdi is a unique feature of Loktak Lake. The Phumdi is ordinary swamp. It consists of thick mat of soil, humus, and a dead piece even in different stages of decay. Upon piece, rich grasses grow up to a height of 5 meters. Next slide, please. And this lake have a great importance value in regard to physical, economic, ecological, and social benefit. They operate as a reservoir of storing water and filter it for recharging groundwater storage. Ecologically, they uphold a large range of all forms of such as mammals, migratory birds, reptiles, amphibians, fish, and many other plant species. Economically, they are endowed with a vast variety of valuable commodities, and socially, a vast number of people carry on their livelihood by making use of them. Next slide.
Hello.
Hello. Am I audible? Yeah. The study we see some change in the land pattern in and around the Loktak Lake because due to the degradation and encroachment in and around the Loktak Lake. The land pattern, there has been a tremendous change in the land pattern and leading to the shrinkage, leading to the diminishing size of this Loktak Lake. Next slide. And this Loktak Lake is confronted with two front challenges. The one challenge is the construction of one Itai Barrage. This Itai Barrage is a project of the National Hydroelectric Power Project. The pressure it gives to the Loktak Lake is a very tremendous. When in order to harness the energy, the electricity from this project, some level of water level is to be kept at the Loktak Lake. So it makes inundated only in and around Loktak Lake areas. So it deprives the livelihood of the people residing there. And the second issue is the sewage was flooded coming from the Imphal town, because there is one river called Namun River. It comes through the Imphal River. Due to the urbanization, many wastes and pollutants brings by this brought by this Namun River and dumped into Loktak Lake. And another is with infestation. The Phumdi. No, I explained earlier. The Phumdi is yeah, right. Next slide, please. Next slide, please. Am I audible? Next slide, please. Next slide, please. Only when someone conclusion part is left to be explained, is to be shared. I think it is almost almost done. I think. from my part. Thank you all. Please carry on, ma'am. So, next slide, please. We come next slide, ma'am. Yeah. Next slide. Sir. It is obvious that traditional measures. Now we come to the concluding part of my presentation. Now it is almost evident that the traditional measures and others, others ways and means to conserve and to preserve this, the floating lake, remain inadequate. All traditional and other methods need to be relooked again, and however, improve, at the same time, we need to improve the living conditions and the livelihood and other alternative livelihoods of the people in and around the Loktak Lake, so that the posting of the Shanghai, the they are available here in Manipur, the posting and easily tracking is to be curtailed, is to be stopped, and we need to have affordable primary health care system, electricity, affordable prices, and these are the issues the confronting with us still now. Now I come to the end of this presentation. Thank you, ma'am. Thank you all.
Now I would like to invite our next speaker, Ms. from Department of Home Science, University of Rajasthan, Jaipur. Ms. Manju, Department of Home Science, University of Rajasthan.
Good afternoon, everyone. I'm Manju from Department of Home Science, and I'm going to present on the topic Role of Climate Smart Agriculture in Achieving Food Security. Next slide, please. The global challenges of the 21st centuries, if we describe them, they are mainly growing global population, changing dietary patterns of the people are changing day by day, food security, agriculture's extreme vulnerability to the climate change, and high greenhouse gas emissions in the agriculture sector. So these are the basic global challenges of this 21st century. Next slide, please. And how this climate change is impacting agriculture and food production. If we see the impact of climate change, it is estimated that yield loss due to climate change can be up to 35% for rice, 20% for wheat, 50% for sorghum, 13% for the barley, and 16% for the maize, depending on the location, future climatic scenario, and projected years. Then climate change and climate variability are emerging as critical challenges for global food security, particularly in underdeveloped and developing economies, because the climate change is majorly affecting the food grain production. And if we see the case of South Asia, it is one of the most densely populated regions in the world and is among the most vulnerable to climate change and climatic variability. This can have major consequences on food security, poverty, and other developmental goals in the absence of adaptation and mitigation. Next slide, please. So, climate smart agriculture is the solution of all to overcome this problem, and it is not a new production system. It is an approach for transforming and reorienting agricultural production systems and food value chains so that they can support sustainable development and can ensure food security under climate change. Climate smart agriculture provides the means to help stakeholders at all levels identify agricultural strategies suitable to their local conditions. Next slide, please. The main objectives of climate smart agriculture are sustainably increase agricultural productivity and income of agricultural producers, adapt and build resilience to climate change, strengthen the capacities of agricultural communities to adapt to the impact of climate change, and reduce or remove greenhouse gas emissions wherever possible. And to reach these objectives, agriculture production systems and food systems will need to use natural resources and other inputs more efficiently and become more resilient to change. Transformations will need to be made in the field at all stages of the food value chain and in policy making also, processes at the local, national and at international level also. Next slide, please. So how do we implement the climate smart agriculture? It can be implemented through expanding the evidence base, supporting the enabling policy framework, having strengthening national and local institutions, and enhancing financing options to have better technologies which are more climate resilient and more sustainable. Next slide, please. There are some identified technologies and practices which can be climate smart and which can be suitable to the agriculture to make it more convenient and more productive. They are first is cropping system improvement. It can be done through improved crop varieties, diversification, crop rotation and integration of legumes, integrated nutrient management which can be like green manure, compost and site specific nutrient management, resource conservation. One of the example is zero or minimum tillage, and then precision water management like planting crops in bed, laser land leveling, mulching with crop residues and agroforestry. Next slide, please. Next slide, please. Thank you.
We are facing some technical issues at our end, so we'll begin with the session after 5 minutes. We'll have a break of 5 minutes and we'll start the session again after 5 minutes. Now I would like to invite our next speaker, Mr. Debashish Sahu from Department of Biotechnology, CSVTU, Bhilai, Chhattisgarh, India.
Thank you, ma'am. So my research topic is biomedical activity of azotides by leaf extract and endophytic fungus associated from cypress species and rima species, along with their potential for biotechnological prospect. Next slide, please. So plants have played an important role in bioremediation of xenobiotics. So some of the xenobiotics for remedial activities that has been already established are heavy metals, azotides, agricultural products such as fertilizers, pesticides, and insecticides, as well as some hydrocarbons. But the drawback is that they have proved to be somewhat successful to some extent. But in respect, they have some limitation to the bioremedical activity with respect to the efficiency. Next slide, please. So here are the two plant species that has been used for bioremediation of azotide. One is Cyperus rotundus, another one is Remax dentadus. These are the two plants that has been isolated and found in elaborate quantity nearer to the pond that contains azotides. Next. So some of the first step was processing of the plant sample. The whole plant was collected and sedoed for 18 to 10 days. Where the moisture content is reduced to less than 10%, the dried plant was then milled into powder, and the powder was then shipped to get a powder form of uniform size, and this was kept in the zipper pouch for further analysis. For the extract preparation, the plant material and the solvents were taken in the ratio of 1 gram to 20 ml, and the sample solvent mixture was kept in 48 hours in second condition at room temperature. After 48 hours, the mixture was then filtered and the filtrate was collected. The filtrate was then desiccated by using literature evaporator to get the condensed extract. So this desiccated extract was again dissolved in DMSO for further analysis. Next. Previous slide, please. Previous slide. The solvent used for the extract preparation, different types of solvent were chosen for that. One is polar solvent that contains water, acidified water for the acidified water containing water and HCl in the ratio of 50:40:2, and intermediate polarity hydroalcoholic extract, ethyl alcohol 55 to 45%, ethyl alcohol 100%, and acetone 100%. So non-polar solvent were not used as because due to the immiscibility with water. The conditions for the extra preparation were pH as per the solvent was used. The temperature of the room temperature of varying from 32 to 70 centigrade. Time was for 72 hours, and the secking speed of the secking parameter was 50 rpm. Next. So for preparation of esterified extract from the endophytic fungus associated with the leaf sample of the plants. So first of all, metabolites were isolated by culturing the endophytic fungus in potato dextrose drop. It is supplemented with 0.1% chloramphenicol antibiotic. So this antibiotic was used to delimit the bacterial growth. So some of the fermentation parameters used for production of metabolite were 5.9, pH, 30 degrees centigrade, and the speed of 70 rpm. So after incubation of almost 10 days, the broth was then filtered by the water number 1 filter paper, and the filtrate was collected for further processing. Again, the filtrate was separated from metabolite extraction by ethyl acetate. Its separation was carried out in separating funnel. So in this case, the water and ethyl acetate forms two separate bilayer, as you can see in the figure. And the upper organic layer with the ethyl acetate layer was extracted and isolated. And it was again desiccated by rotary evaporator or vacuum evaporator, and it was reconstituted by acetonitrile 100%. Next slide. So some of the codes that were being given were Cyprus, I have coded at T, Rumex was coded at E, and some of the solvent code were water 100%, esterified water, water to STL 50-40, hydroalcoholic extract, ethyl alcohol, and 55 to 45, ethyl alcohol 100%, acetonitrile indified, acetonitrile that is been reconstituted from the ethyl acetate layer or acetic acid organic layer for endophytic fungus, and sixth one is the acetone layer that is 100%. Some of the dyes that were present, that were used for the assay were 0.1%, and here methyl red, methyl yellow, rhodamine, and Congo red. These are the four dyes that have been used for the assay of azurite reduction. These are the different azudites, and the concentration was for initial screening was 0.1%. The reduction of different dyes and the coloration of assay were by target plant extract were defined by this formulation. Percentage of degradation equal to absorbance of control minus absorbance of test by absorbance of control into 100. This was analyzed by visible spectrophotometry. Next. Next slide, please. As from the graph, you can see that the methyl red, methyl yellow, rhodamine, and Congo red were used for the assay have been degraded, and whatever you see on the graph on the upper side of the graph, these are the percentage of degradation has been elaborated from the equation of percentage of degradation. You can see that the polar solvents, that is polar solvent of both of Rotundus and Cyprus. So both have better activity against methyl red, methyl yellow, and Congo red. And coming to the fact that E5, that is the acetonitrile extract or esterified extract of endophytic fungus, you can see it has also given positive for degradation of methyl red, methyl yellow, and Congo red. So here, rhodamine has a more or less least effective. Means my extract and the endophytic fungus that have been extracted are least effective against the rhodamine, whereas most effective was against Congo red, methyl yellow, and to some extent to methyl red. Next slide, please. So this is the screening of the silver nanoparticle, and here only the hydroalcoholic extract from Rumex and the esterified extract from endophytic fungus that is also isolated from Rumex have given the positive for silver nitrate, silver nanoparticles from silver nitrate solution. Here you can see 1 millimolar silver solution initially was taken, and the leaf extract was taken 0.5 ml, and after 36 hours, the color of the solution changed from the initial color to reddish brown or brownish red color, giving the indication that the formation of the silver nanoparticles. Next slide, please. And the silver nanoparticle was again purified by continuous centrifugation and extraction procedures. Finally, the pellets were reconstituted by double distilled water. In order to find out the characterization of silver nanoparticles, so the reconstituted pellet was taken and 4 part of distilled water was taken, and it was kept in ambient temperature for overnight. And the analysis was carried out by UV visible region. If we found the absorbance peak at 420 to 450 nanometer, that correspond to the formation of silver nanoparticle. And yes, I have found out some of the higher spectral analysis or peaks in between 420 to 450 nanometers, for indicating that silver nanoparticle has been produced from the plant extracts. Next. So getting into the morphology by TEM, the morphology shows that the silver nanoparticle that are produced by the extract and the endophytic fungus have been highly variable, giving the indication that the silver nanoparticle have been produced, the aggregates of the silver nanoparticle that is produced is in between the range of 50 nanometer to 20 nanometer in case of hydroalcoholic extract and 20 to 70 nanometer from esterified extract. Next. So in case of identification of the endophytic fungus, so we have gone for the co-culture on the STA media, as well as the LCB-APS media or the lactophenol cotton blue staining, where you get that endophytic fungus isolated and was identified to be Cladosporium species. Again, qualitative analysis of secondary metabolite was for hydroalcoholic extract, and endophytic fungus was also determined, where we found out that those extract contains different chemical compounds such as alkaloid, flavonoid, phenolic, carbohydrate, proteins, glycosides, saponinoids, quinone, triterpenes, and sequitotropins and tannins for hydroalcoholic extract, whereas the eutrophied extract of endophytic fungus contained alkaloid, flavonoid, phenolic, tannin, coumarin, resin, proteins, and glycoside. So these are the different phytonutrients, phytoconstituents that are present in the secondary metabolites. Next. So the observation is that afterwards, some of the observations were also carried out for extract as well as the silver nanoparticle, where we found that the combined effect of extract and AGNP or the silver nanoparticle have enhanced activity in the reduction by varying from 10 to 25%. So my conclusion is that limited study has been proposed for plant extract and endophytic fungus as potential for environmental bioremediation. So present study proved promising candidate for bioremediation. So future studies shall indicate identification of both biomolecule and phytomolecules responsible for bioremedial activity, along with integration of nanotechnology with plant-based remedial activity, which will give new insight to faster, cleaner, efficient, quality-based, environmental friendly, safer and non-toxicological alternative as compared to other techniques that is currently available. Next slide. Yeah, thank you.
Next, I would like to call upon our next speaker, Mr. Subhash Chandra Arora, Faculty of Management Studies, Gurukul Kangra University, Haridwar, Uttarakhand.
Yes,
Mr. Subhash Chandra Arora.
yes, am I audible?
Yes, sir, you are audible.
Okay, thank you. Okay, so good afternoon to all the respected professors, faculty members, organizers of this conference, and all the members present over this soft board. I would like to thank Indira Gandhi Center for Human Ecology, Environmental and Population Studies, University of Rajasthan, for organizing this international conference and giving me an opportunity to express and share my research topic titled as Rethinking the Future of Mobility in the Indian Market and Application of Extended Diffusion of Innovation Framework. Starting with the introduction part. Next slide, please. Starting with the introductory part, as we all know that pollution of the environment is currently a global concern. Advancement in technology is making the lives of humans better, but has also disrupted the ecosystem to a great extent, thereby putting a variety of species of plants and animals to a great risk of extinction. This global warming is also threatening the social and economic well-being of individuals. Although there are many industries that are contributing to the global climate change, but it is primarily the transportation sector that is becoming a cause of concern. This sector includes a variety of activities, but primarily it is the road transportation sector that is responsible for almost 70 to 75% of the total transportation carbon dioxide emissions. The demand for vehicles is increasing on a year-on-year basis due to various reasons like increased economic activity, increasing population, and car remaining no more a luxury for a significant proportion of the population. And this increasing demand is leading to various greenhouse gas emissions such as carbon dioxide, nitrous oxide, sulfur dioxide, and other tiny particulate matter into the environment. Therefore, what we need is a more sustainable form of vehicular technology that is presently available in the market, which is nothing but the electric vehicles. Electric vehicles are being defined as vehicles that use electricity as fuel for propulsion. Instead of using internal combustion engines, they use battery technology to run the onboard motors. Countries and companies all around the world are aggressively promoting this new technology so as to reduce the global carbon dioxide emissions target, plus to achieve the intended targets made under the Paris Climate Agreement 2015. But despite a series of measures being undertaken, the overall market share or the global market share of this vehicle, of this technology, is just 2.6%, if we talk about India, in 2019, only 1.56 lakh electric vehicles were sold, out of which only 3,400 were electric cars. Next slide, please. Next slide. So therefore, this study was being undertaken by me to previous slide. So this study was being undertaken to know the intentions of consumers as to what they feel about this vehicular technology. The variables were identified in this research were being undertaken by a very famous paper or from a very famous model being developed by Rogers in 2003. He identified 5 variables that play their role in influencing the purchase decision of consumers in case of any product. Besides the variables that he has suggested, the author has suggested one more variable was added so as to explain or to increase the explanatory power of the model that has been used here. Next slide. So this is the conceptual model that has been developed based on the literature that I have read during the last few months. The hypothesis being developed in this model related advantages positively affect the intention of consumers to adopt electric vehicles in the near future. Simultaneously, it is the compatibility which positively affects complexity of using this technology or complexity of using this vehicle negatively affects the intention to purchase it. The role of triability, observability, and the attitude that consumer holds towards this product is also hypothesized to be positively related to the intentions to adopt the electric vehicles. Next slide. So this was the literature that these were the few papers that were being studied. To quote one very relevant paper published in 2011 by John Johnson, 1766 responses were. There is a huge difference between the adopters and non-adopters who have purchased this vehicle in the latest year. Next slide, ma'am. So talking about the research gap, although there are many studies that have used different theoretical frameworks to look into these various factors that explain the purchase intention of customers, but most of these studies were restricted to select countries, especially the European and other Western countries. Thus, what it is that encourages consumers to purchase or adopt electric vehicles in the Indian context comes to the attention in this research. It can be pointed out that the literature related to electric vehicles is still inadequate or rather fragmented, given the consideration that the industry is still emerging in both national and international markets. Next slide, ma'am. So next slide. Previous slide. Previous. So the data was collected from a particular city, that is New Delhi, and the responses from 236 respondents was collected, and the demographic profile of this survey has been shown in front of you. Next slide. So the variables that we have identified has revealed factor loadings, Cronbach alpha value, and composite reliability of greater than 0.7, which denotes that the instrument that was used to collect responses from the consumers was consistent in all its forms. The last column of average variance expected denotes that the items that were being mentioned in that questionnaire were not closely related to each other. Therefore, a lower value of a value of less than 0.7 was being considered as sufficient to establish that there is adequate variance between all of the items that were being mentioned. Next slide. So the results were derived from using a software called as AMOS software, and the result of this questionnaire revealed that it is the attitude that significantly impacts the intention of the consumers, followed by the related advantages. Although complexity played its role in this survey, but the amount is not significant enough to reveal that the consumers feel negative about this vehicular technology. Next slide. So it can be concluded that This research work will suggest policymakers and other stakeholders as to what can be done in order to improve the penetration of these green vehicles in the Indian market, especially from a consumer perspective. This study would provide useful indicators regarding the positioning process and the channelizing of resources and efforts into the right direction, and which consumers to target and which markets to explore first. Thank you, ma'am. Next.
Now I would like to call upon our next participant, Vishwajit Vaishnav from Department of Botany, Tripura University, India. Mr. Vishwajit Vaishnav.
Am I audible, ma'am?
Yes, sir, you're audible. You can start with your presentation.
Good afternoon, respected chairperson and other dignitaries and participants. Myself, Vishwajit Vaishnav from Tripura University, and I am presenting my research work, and the title is Present Status of Orchid Species Diversity and Their Distribution in Tripura, Northeast India. Next slide. The introduction. Orchids has most amazing flowers on earth, appertain to the family Orchidaceae. Orchidaceae are diverse and widespread family of angiosperms and supposed to be the highest evolved twin monopod, occurring 28,000 currently accepted species, including 763 genera. They are precious for their diverse size, shapes, forms, fragrance, color, and their attractiveness of flowers. They comprise approximately 8% of all vascular plants, grow in almost all terrestrial habitats except in dry deserts, and are native to all continents except Antarctica. According to Mishra in 2007, there are about 1,331 species distributed in 186 genera have been reported from India. Next slide, please. Northeast India is rich in orchid bioresource. Northeast India is a mega diversity center comprises of 7 states. This region of India is rich in biodiversity due to high rainfall and plenty of sunlight. This region consists of about 72.8% of the country's orchids, occurring 159 orchid genera comprising of 870 species. Tripura is also rich in orchid diversity, where 66 species of 77 genera have been reported earlier by Kotoki et al. But in the state flora by D.B. Dev in 1983 mentioned 31 orchid species belonging to 22 genera. Since the time of the work done by Dev in 1983, no details survey was conducted in Tripura for the orchid flora. Hence, my objectives of this study is to find out the actual orchid diversity and their present distribution status in Tripura. Next slide. Study area. My study area is my state, Tripura. This state also further divided into 8 small districts, and the Tripura exhibit typically tropical dry and wet climate with a high level of humidity and heavy rainfall. The rainy and moist environment of the state supports a diversified flora, including rich orchid diversity. This figure shows the map of Tripura, pointed with several geographical location which are the different habitat distribution of orchid species of Tripura. Next slide. Now the materials methods. Rapid field survey were made throughout the state in different districts. During field exploration, we also collect several orchid species. To identify the collected specimen, we use several taxonomic description or suitable literatures and relevant taxonomic study. The specimens of herbarium specimens are deposited in the Department of Botany of this university. Next slide. Now the results. We have observed or and also collect 40 orchid species from Tripura belongs to 25 genera. Out of 40 orchid species, 31 species are epiphyte and 9 species are terrestrial. The genus Dendrobium is the highest species containing genus and thus showing highest diversified genera in orchid genera in Tripura. Some of the epiphytic orchids of Tripura are Acampy primorsa, Erids odorata, Cymbidium aloefolium, Chrysostoma paniculata, Palantheria insectivora, etc. And the terrestrial orchids which are exhibiting Tripura, such as Heteria epinis, Liferis odorata, Orundina graminifolia, etc. Next slide. These are the figures showing field activity during field survey. Next slide. This figure, this slide shows several orchid species. Picture of several orchid species found in Tripura. Number 2 is Bulbophyllum lobi, and number 8 is Dendrobium densiflorum. Number 9 is Dendrobium fimbriatum. Next slide. This slide also shows some of the figure of the some of the orchid species. Number 2 is a terrestrial orchid species, Geodorum densiflorum. Number 3 also a terrestrial orchid, Heteria affinis, and number 8 is Rhynchostylis retosa. Next slide. Here also some of the picture showing number 3 shows Orundina graminifolia. Number 9 is Gooderia procera. Next slide. Now the conclusion. This study reveals 40 orchid species in Tripura, and future studies may include more genus and species in the orchid flora of Tripura. 34 orchid species are under conserved in shade net house in this university. Majority of the species are distributed in moist and semi-deciduous forest spaces of Tripura. Some orchids are commonly distributed throughout the state, whereas some are observed on the roadside forestry on tree trunk. The high species diversity found in north districts of Jampu Hill ranges and other districts of Tripura explored with less species diversity. There is a at last there is a urgent need of conservation of this diversity due to habitat destruction to check the population extinction. Thank you.
Now I would like to invite our next presenter, Ms. Shivani Sharma from Department of Environmental Sciences, IIS University, Jaipur.
Yes, ma'am. Am I audible, ma'am?
Yes, yes, Shivani, you can start with your presentation.
Okay. Good afternoon, everyone. Good afternoon.
Please put on your video.
Okay, ma'am. Good afternoon, everyone. Here I am going to present my work on the topic concentration of ultrafine particulate matter in residential area of urban site. Next slide. As we know, harmful gases and the presence of certain physical, chemical, and biological agents in the environment due to natural and anthropogenic activities which affect the living beings, including plant, animals, and human beings. It is caused by natural processes such as volcanic eruptions and wildfire, pollen grain, dust storm, bacteria, microorganisms, etc. And by anthropogenic activities, it is caused by construction, mining, transportation, industrial work, agriculture, smelting, etc. Air pollution can be studied as an indoor air pollution, ambient air pollution. Next slide, please. Hello, ma'am. Am I audible? Excuse me, ma'am, am I audible?
Yes, Shivani, you are audible.
Ma'am,
You can
please
go
change.
ahead.
Ma'am, next slide, please. Pollutants. An air pollutant is a substance in the air that can adverse effect on humans and the ecosystem. The substance can be solid particle, liquid droplets, or gases. A pollutant can be natural origin or man-made. These are of two types, primary air pollutants and secondary air pollutants. Primary air pollutants are those which emit directly into the environment, such as ash from a volcanic eruption. Major primary pollutants include particulate matter, sulfur dioxide, nitrogen dioxide, volatile organic compounds, carbon monoxide, and lead. Next slide, please. Secondary air pollutants are those which are not emitted directly into the environment, rather they are formed in the air when primary pollutant react or interact. An important example of secondary pollutant is ground level ozone, one of the many secondary pollutants that make up photochemical smoke. Next slide, please. These are the various health impacts of pollutants. For particulate matter causes respiratory and cardiovascular illness. Sulfur dioxide again causes respiratory function. Nitrogen dioxide also irritate the lungs and lower resistance to respiratory infection. Ozone causes asthma and allergic triggers. Radon also leading cause of lung cancer in non-smokers. Next slide, please. So the objective of my study is to evaluate the mass concentration of ambient ultrafine particulate matter in residential area and to estimate the concentration of gaseous pollutants SO2 and NO2. Next slide, please. Due to the high proximity to highway, the area experience high traffic volume. The emissions of harmful gases is one of the major concerns to study. In this area, the traffic light also makes it shopping malls, due to which dispersion of pollutants may not occur. So sampling I have been collected from December to March, thrice in a week. Next slide, please. Next slide, ma'am. These are the samplers for PM1. Yeah. For PM1, the sampling duration was conducted for 24 hours using PTFE filter paper with 37 mm diameter, and for gaseous attachment, SO2 was conducted for 8 absorbing bins. Next slide, please. The air samples for various parameters have been analyzed by the method given below. For PM1 gravimetrically, SO2 was analyzed by Jacob and Harsha method, NO2 was analyzed by sodium arsenide method. Next slide, please. The results of the as we see in the results, that the concentration of PM1 was found higher in the month January, and similarly SO2 and NO2 are also higher, but they are in the permissible limit. Next slide, please. So the conclusion, the study conducted at the Mansarovar area to evaluate the ambient air quality at suburban residential area revealed that the major contaminants of air are PM1, SO2 and NO2. The sources are the vehicular emissions, road traffic and garbage burning at nearby highway and residential areas. Further degraded due to the construction of tall buildings in the nearby areas has dispersed the dispersion of pollutants, worsening the problems of poor air quality. Next, the concentration of PM1 is much higher. This is concern because such levels of air pollutants are harmful to the health and needs to immediate action to be taken. On the SO2 and NO2 are under permissible limit as prescribed by the CPCB, that is 80 microgram per meter cube.
Now I would like to call upon the next participant, Harsha Parwani from Department of Environmental Sciences, IIS University, Jaipur.
Good afternoon, everyone. Am I audible, ma'am?
Yes, you are audible. You can begin with your presentation.
Okay, ma'am. Please share the presentation.
Shravan. Are the slides visible to you?
No, ma'am.
You can begin with your presentation, Harsha.
Ma'am, slides are not visible. Yes, ma'am. Yes, ma'am. Now they are visible. Good afternoon, everyone. I'm going to present on indoor air pollutants and their health effects. These are my outlines. Next slide. Indoor air quality is the air inside the building, including the concentration of pollutants and their thermal conditions that affect health and comfort of an occupant. The contribution of indoor air pollutants to diseases in India is approximately 3.8 million. Among various other air pollutants, fine particulate matters have been found to have profound effect on human health. Its diameter is approximately 1/50th of the width of the human ear. Basically, particle size plays an important role in accurate and reliable assessment of impact on health because the degree of respiratory penetration and retention of particulate matter is a direct function of its aerodynamic diameter. Next slide, please. I have basically taken the indoor air pollutants at schools. As growing children are spending more time at school, they are more prone to indoor air pollutants, as the exposure to indoor air pollutants can interfere with their learning abilities. Symptoms associated with indoor air pollutants are nausea, fatigue, drowsiness, etc. This leads to increase in absenteeism in the school. Next slide, please. Why to focus on indoor air quality? Because to reduce outdoor air pollution, to change in construction practices, like the materials should be permeable to air, prefabricated homes, new heating methods should be used, energy conservation methods should be used. Next slide, ma'am. These are some of the sources of indoor air pollutants. Outdoor sources includes traffic, waste burning, and etc. Indoor sources includes construction material, disinfecting products, etc. Next slide, ma'am. These are some of the health effects which are caused due to indoor air pollutants in children. Impacts on respiratory system includes acute effects includes mucous membrane irritation, coughing, wheezing, increased responsiveness of the respiratory tracts to allergens, increased acute respiratory morbidity. Chronic effects includes decreased lung function and COPD. Next slide, ma'am. These are the impacts on cardiovascular system, which involves increase in blood pressure and heart rate and stress hormones. Now, the effect of pollutants on immune system includes infections, absenteeism from school due to sore throat, cough, and cold, and increased levels of oxidative stress and inflammation. Next, impact on central nervous system. Its acute effects include headache, fatigue, nausea, and impaired task performance. Next slide, ma'am. Chronic effects include learning disabilities, memory functions, reading comprehensions, changes in gray and white matter in the brain, and prenatal and early childhood exposure, that is autism in autism. Next is cancer inducing effect include children leukemia, and endocrine impact includes impairment of reproductive system, disorders in brain, and contribution to later diabetes, obesity, and thyroidism. Next slide, ma'am. These are some of the effects of sick building syndrome and building related illness. Sick building syndrome, the symptoms are clearly visible when we are away from the building, whereas in building related illness, symptoms are not visible after leaving the building. Sick building syndrome symptoms include headache, fatigue, irritation in eyes, nose, and throat. And building related illness include asthma, cold, flu, allergic reactions, etc. Next slide, ma'am. These are the some of the management of indoor air quality in schools. That is, proper cleanliness should be maintained at schools. Arrangement of furnitures, workspace, overcrowding should be avoided. Children should not be seated in direct sunlight. Noise should be controlled. This helps in improving the learner's performance. Next slide, ma'am. These are the conclusions of the indoor air quality. Good indoor air quality is a guarantee of comfort, health, and safety. Growing children are very sensitive to hazardous chemicals. Exposure to indoor air quality in schools can keep back learners' task performance. Material with low emission, adequate ventilation, and cleaning methods, temperature, humidity, and mold control all take part in improving indoor air quality. It is an essential component of healthy school environment. Next slide. Thank you, ma'am.
Now, I would like to call Gaurav Kotiar from Sri Guru Nam Rai University, Dehradun, Uttarakhand. Gaurav Kotiar. Gaurav Kotiar, are you here with us?
I am on.
So are you sharing your screen? Will you be sharing your screen, sir?
Ma'am, there is some network problem, ma'am.
Okay, so till then I'll call another participant till you resolve the issue. Okay?
Okay, ma'am.
Now I would like to call Dr. Chandpal Singh from Department of Botany, University of Rajasthan, Jaipur.
Thank you. Could you please allow me to share my PPT?
Yes, sir. We are sharing it. Is the screen visible to you?
I would like to share from my side.
Okay.
Yeah.
Now you can share your screen, sir.
Yes. Okay. Hope it is visible.
Yes, sir.
Okay, respected chairperson, Professor Subhash Chandra ji, organizers and dear participants. Today, I'm presenting piece of my research work that is dealing with microRNA and its role in host pathogen interaction using the model silkworm and its virus. So microRNA, these are small uncoded RNA length around 21 nucleotides, and they are processed from a stem loop structure, and they are widely conserved in higher eukaryotes. And currently, more than 48,000 microRNA has been identified, which are regulating more than 60% of the human genes. So that reflects the importance of microRNA. This is the basic biogenesis pathway of microRNA in animal, or also conserved in insects. So suppose this is genome transcribed by RNA polymerase II, having a transcript which has self-complementary sequences due to which stem loop structures are formed, and these are recognized by an enzyme complex containing DROSHA and PASHA, that is a RNase third enzyme. It processes this primary miRNA transcript into a precursor miRNA. So precursor miRNA around is length is around 60 to 90 nucleotide. Then it is transported by exportin 5 using the energy from RAN-GTP into the cytoplasm, where further this precursor microRNA is processed by dicer into mature miRNA duplex and further loaded onto a complex that is called RNA-induced silencing complex. For microRNA, it is called MIS, where a protein called argonaute is a main effector molecule, which is having property of helicase. So, sorry, not helicase, RNase H activity. So it can cleave the RNA in a duplex. But that depends on the complementary between microRNA and the target mRNA, as you can see here. So these are possible outcomes. mRNA deadenylation means poly may be removed. Directly degradation if it is perfect complementary, and repression means protein synthesis will be suppressed. Okay, because our case is dealing with the virus, so why virus require microRNA? Because there are certain advantages, because they occupy very less space in the genome, and they can regulate their own gene and host gene, as well as they can sustain the viral life cycle in the host by regulating its own genes for a longer period. So it's like a persistence. This is a diagrammatic representation how microRNA can play a role in the host pathogen interaction. So DNA virus pathway is totally known. For RNA virus, this is not fully known how RNA virus can produce a microRNA. So suppose these are mature miRNA using the same pathway which is present in the host. They are regulating viral mRNA, they are regulating host mRNA. So the possibilities are there with the microRNA. They can regulate both type of genes. So using this microRNA, we did some work in the insect virus interactions using Bombyx mori, that is mulberry silkworm, and its pathogen Bombyx mori nuclear polyhedrosis virus, that is BMNPV. So first objective in this was to identify microRNA from the virus. Why it is important to study this virus? Because you can see here it contribute to more than 50% loss, means directly loss to the silkworm industry, sericulture industry. So using the sequencing, the small RNA sequencing bioinformatic approach, we identified finally 4 microRNA. This is the pipeline of bioinformatic approach, and this is the virus genome showing the region from which microRNA are derived. This northern blot image present, you can see in fat body. Fat body is like a liver for insects, and midgut, you can see the expression is there. As I told that microRNA are small, so their size is 19 to 28 nucleotide. Here we got around 22, and only for 1 microRNA we also received the miRNA. Because this was the pioneer study in India, so we again checked using the different method, stem loop PCR method. For their expression, you can see expression is found in fat body and midgut, which were infected with the virus. However, there was no infection in the control, which were having no infection of virus. Similarly, another approach was used, poly-A-tailed RT-PCR. You can see their expression was thoroughly validated in the midgut. Then, after identifying these microRNA, we analyzed one of the microRNA's function in the virus gene regulation. So we chose the BMNP MIR3 microRNA. These are named based on their microRNA nomenclature and virus. So BMNP from the virus and MIR3 is the number of microRNA. It found to regulate DNA binding protein P6.9 3' UTR. So UTR means that is untranslated region in the mRNA at the 3' side where microRNA generally found, binding generally observed. So after predicting the site on this mRNA, we validated its interest using the luciferase assay. In luciferase assay, what we do, we make a construct in that target sequence is inserted, and both the microRNA and this vector is transfected in the cells. Then we analyze the expression of luciferase. You can see more than 50% repression was observed when microRNA specifically interacted with the P6.9. However, there was no change with the unrelated microRNA with unrelated target, etc. Then further we validated its expression profile in the tissue at different time level. So you can see this is infected fat body, infected midgut, silk gland, and these are the silkworm, means culture cells. These are cells which are cultured out of the silkworm body. So these are ovary cells actually. So you can see microRNA is heavily expressed in the initial stage at 12 HPI and decreasing successively. And it is already known that this virus won't go to the silk gland. And the reason exactly not known. What is the reason why this microRNA won't, or this virus won't infect the silk gland? Okay. After validating the interaction of this P6.9 gene and microRNA, I got an idea that because this is a late gene, whether this is targeting only the single gene or many more viral late genes are also targeted by this microRNA, then I predicted targets for this microRNA. These are the interaction energy for these microRNA targets. You can see this is their respective names and known functions. Then generally two approaches are used for validating microRNA functions. One is blocking the microRNA, another is overexpression. So this first experiment shows blocking of microRNA, which is blocked by the LNA, locked nucleic acid. 3 means microRNA 3. So whenever microRNA is blocked, you can see the genes which is targeted by this microRNA are overexpressing, means the hindrance by microRNA is removed. Then for different time level and for only P6.9, I analyzed using the real-time PCR. So you can see more than 25-fold increases there when microRNA is blocked by a oligonucleotide. In the reverse case, when microRNA is overexpressed, you can see the target expression goes down except this gene. So later I removed this target from the list. Then this is analyzed for the viral load in the silkworm tissue and viral load using the DNA level. So this DNA level of virus is measured at different time levels. So you can see it is decreasing. In another approach, virus level was analyzed when microRNA was blocked. So microRNA is blocked, you can see virus is proliferating, means this microRNA is required to regulate the virus. So it is a very interesting case. Virus using its own microRNA to regulate its own gene to regulate the viral titer. Then using the viral bodies, you can see when microRNA is blocked, virus is proliferating. To have an exact idea why this microRNA is suppressing viral genes and viral load, we analyze the expression of an antimicrobial protein called gloverin-1. So whenever there is a virus infection, this gene is induced. So you can see interestingly, when this microRNA is overexpressed, virus level is very less, means the gloverin level is very less because virus is not recognized by the system due to this microRNA However, in the reverse case, when LNA is blocking microRNA, so this microRNA is not regulating the viral genes. You can see virus is proliferating. So system can recognize the virus and produce a immune response against it. So based on these all results, we propose a model where this microRNA is synthesized and regulating the early expression of late genes. So this is very interesting. As we know, some genes are in virus are early, very early, late, very late. Who can regulate them? So this is the case of direct regulation of viral late genes early expression by a microRNA. So by which host cannot recognize the signal of virus, and later it can enjoy the freedom using the viral load. When viral load is more, host cannot do anything. So it will surrender. Only in the initial stage when virus is low, host can recognize the pathogen particles and can produce the immune response. So overall summary is we identified 4 microRNA and characterize the function of one of the microRNA. And this has a potential application in using insect as a virus as a biocontrol agent, as well as producing the resistant silkworm against the virus. Thank you for your kind attention, and I congratulate organizer for organizing such a wonderful conference. If you have any question, I will be happy to answer your questions. Thank you.
Thank you, sir.
Yeah, thank you.
Now, I would like to call Mr. Gaurav Kotyal from Sri Guru Ram Rai University, Dehradun, Uttarakhand. Gaurav Kotyal. Mr. Gaurav Kotyal. We are audible to you. Okay. Now, I would like to call upon our next participant, Garima Singh from Indira Gandhi Center for Human Ecology, Environmental and Population Studies, University of Rajasthan, Jaipur. Garima?
Yes, ma'am, your voice. Yes, ma'am, your voice is audible to me.
Okay.
Good afternoon, everyone. The topic of my presentation is analysis of the groundwater quality of industrial area of Jaipur. Next slide, please. EIA stands for Environmental Impact Assessment. EIA can be defined as the study to predict the effect of a proposed activity or project on the environment. EIA compares various alternatives for a project and seeks to identify the one which is the best combination of economic and environmental costs and benefits. EIA integrates the environmental concerns in the developmental activities at the time of its initiation. Next. Core values of EIA are integrating, which should be fair and unbiased. It should have utility, that is, it should provide authentic information to the decision makers, and it should be sustainable, that is, it should result in environmental safeguard. Next. Groundwater. Groundwater, as we all know, is the water that exists underground in the saturated zones beneath the land surface. Groundwater may be found almost everywhere. The water table may be deep or shallow and may rise or fall depending upon various factors. However, heavy pumping of the groundwater supplies may cause the groundwater to fall. In the areas where the materials above the aquifer is permeable, the pollutants can sink into the groundwater supplies. Next. Groundwater pollution occurs when the pollutants are released to the ground and make their way into the underground water. Using the polluted groundwater cause hazards to public health through poisoning or spread of the disease. Next. Next slide, please. The area of the current study analysis is the Jhotwada industrial area. The area is located in the northwest part of the Jaipur. It is situated between Jaipur-Delhi bypass Binard Road. As per census 2011, the population of the area is 1,39,506. The area witnesses around 180 industries. Next. The groundwater samples were collected from the industrial area within the 10-kilometer radius and were analyzed for the following parameters as pH, electrical conductivity, total dissolved solids, chloride, fluoride, residual chlorine. Next. Acidity and alkalinity. Next slide. The results revealed considerable variation in the chemical composition of the water samples. Most of the collected water samples were seen to have fluoride and chloride beyond the permissible limits. The industries released their wastewater in the adjoining areas without any treatment. This can be seen as a possible reason for the contamination of the groundwater. Next. Thank you.
Now, I would like to introduce Professor Gyan Singh Shekhawat.
Hello.
Professor Shekhawat is working as professor in Department of Botany, Jai Narayan Vyas University, Jodhpur. He has been awarded by INSA International Exchange Fellowship, Young Research Award, Professor Smuti Gold Medal Award by Indian Botanical Society. Dr. Shekhawat has completed several research projects funded by different agencies like UGC, SERV, DST, Delhi, and CSIR. He has published 2 books, 11 book chapters, and 65 research papers in journals of national and international companies. Presently, he is working on hemooxygenase 1, HO1, nitric oxide signaling in plants and in vitro production of medically potent metabolites. Now, we'll welcome you, sir. Over to you, sir.
Hello. Am I also audible?
Yes, sir. You are audible, sir.
Thank you, ज्ञान सिंह जी. Thank you. आपके आने से मेरा दूसरा पीस लैपटॉप से जुड़ा और मैं बिल्कुल ऑडिबल हो गया. Thank you. आप बोलिए ज्ञान सिंह जी. Thank you.
Thank you very much for a generous introduction. And as you know, we are the whole webinar is dealing with such an important issue regarding to the environmental issue. And you know, whenever we have a change in environmental issue, the most important, the producers have a lot of pressure on them. So the producers mean plants, and to deal with this pressure, plant have number of adaptations, changes at the physiological level, changes at the morphological level, and so on. So there are 2 types of the adaptation plant is doing. One at the morphological level and one at the cellular level. Cellular level includes at the physiological level and molecular level. So how we can by this changing environment, by changing temperature is rising, we have the different changing of the pattern of the seasonal pattern. So these all the changes are ultimately comes to the plant and plant how the plant is deal with that. So I'm sharing my screen now so that you can start our presentation. So my presentation today, I have just a brief all the things, because a lot of presentation is going on to these aspects, and how we can do these are the with the plant are the some enzymatic is a very important enzymatic aspect is very important. So we have the number of enzymes through which the plant is deal these such problems. Like we have an APX, we have a catalase, we have a superoxide dismutase. But these are the already explored very well in the different plant species and different plant cells. One enzyme that is very recently identified, which is very interesting also, that is the heme oxygenase. Why this is important? Because this enzyme is ubiquitous in nature. Ubiquitous mean that is present throughout the organism. They started from the cyanobacteria to animal system. That means Homo sapiens sapien. So that is why this enzyme is more important, and that plays number of roles, number of chemical reaction within the cell of cyanobacteria to Homo sapiens. So that makes this enzyme very important. And obviously we know that all the enzymes are the protein, and they have a very specific role in the individual. If we talk about the fungal species, we talk about the bacteria species, and then the lower plant to the higher plant, they have the different variation in that. So when we classify this enzyme, we can classify the two broader category to this enzyme. That is the subfamily 1 and 2, and just directly jump to their individual classification. Then we call it the heme oxygenase 1, 2, 3, and 4. So this is the till date, maybe they have the more subunits of these enzymes into the plant, but in broader category, these are the 4 enzymes which is identified till today, and they have a play different role into the different plant species, different plant tissue, and different plant organs. So this is the basic idea about this plant. Then I have also mentioned, in earlier slide, because this is interesting in that way, because it's present throughout the organism. That is the cyanobacteria to mammals. And then what are the difference between them? So the mechanism of the heme degradation is a common feature to the whole organism. So heme, that means we call that an iron. So the degradation heme oxygenase is throughout the common, but basically they have a diversity in the reaction products. So in the in different animal, they have a different precursor and different reaction material. So that makes this plant very specific to the specific individual species. When we started, because I supposed to mention here that this enzyme is basically identified from the animals, because the animal have the more heme, that means we call the hemoglobin. So hemoglobin have and the RBC is contained the hemoglobin, and which is required to degrade very frequently. So this enzyme is basically degrade the heme into the heme and the porphyrin ring. So these are the basic function of this enzyme. But when we start this work almost a decade back, then it is very important to establish the protocol to identify this enzyme into the plant species, because we try to check its role into the plants. Then we haven't modified the protocol which is present into this. So in this slide, you can see the whole what heme oxygenase is actually doing. So heme is the main unit of the porphyrin, and then this is degraded into the biliverdin 9-alpha, and which is further degraded into the bilirubin. This is the function into the animal cell. And here is also, you can see in the plant system, it has a different role, and during the process of heme reaction, they produce the carbon monoxide and the iron moiety, which is very important. They play a role as a secondary messenger. We discuss in the later slides. So this is the fundamental aspect we have established into the plant cell, how we can calculate the heme oxygenase into the plant cell. So this is then I just brief of the some important aspect of this heme oxygenase into the human cell. So basically, you see the central circle, that is the heme degradation, which is the primary role of the heme oxygenase into the any animal cells, including a human being, a Homo sapiens. Then the next important role, it has the apoptosis suppression. So apoptosis is the very common, phenomenon which is suppressed by the heme oxygenase. Then this heme oxygenase also play as a secondary messenger. That is the second messenger, and which is very interesting role, and we focused in the plant how the heme oxygenase play as a messenger role into the plant system. Then HSP, we all know about the HSP, heat soak protein. So this heme oxygenase also play important role as a heat soak element into the animal cell. This slide is all about the animal cell. And you see, if you put the heme oxygenase, you get the hundreds of paper into the different animal species, and including the Nature, Science, and you name any of the journal, you find the one paper of the heme oxygenase. Maybe the PNAS, maybe the FABS, maybe the JBC, maybe the Science, Nature, Scientific Reports. These are the journal, they cover all the role of heme oxygenase into the animal cell. However, into the case of plant, there is almost no reports of the heme oxygenase. So we started with the collaboration with the University of South, Florida, and then the Southampton, UK. So these are the 3 labs which is working onto the heme oxygenase with the plant, what are the heme oxygenase doing into the plant system. So if we have one paper, the review paper into the JSPB, published in the 2010. So we have summarized the whole role of the heme oxygenase into the plant cell. So one important role as an antioxidant. When the plant into the stress condition, this heme oxygenase is overexpressed and help to the plant to accommodate with the stress. Then very important role, that is the phytochrome chromophore synthesis. I suppose to mention here one thing. You see the evolution. In the case of animal, these enzymes degrade the heme. That means the hemoglobin, the heme is present in the hemoglobin, it's degraded by the heme and it separates the heme moiety and the porphyrin separately. But in the case of plant, it's almost opposite. It helps into the phytochrome chromophore synthesis. Let me explain what is the phytochrome chromophore. Phytochrome chromophore is the unit which is before to the chlorophyll. The chlorophyll is the more complex molecule, but before the chlorophyll, this is the phytochrome chromophore. So this enzyme help into the synthesis of phytochrome chromophore. So you can imagine the trend of evolution. In the one way, this enzyme degrade the heme. In case of plant, it's help into the synthesis of the chlorophyll. So there's a very unique, and one can start their work into different aspects of the heme oxygenase, and any scholar whoever wants to work with the plant and plant cell related issue with the heme oxygenase. So this is a very unique work, which anybody can take up. Very few people are working nowadays. Then signaling component, which is very interesting, and we share some slide, with the signaling, how the heme oxygenase help into the signaling process. And the last, it also have some developmental role. Developmental role, that means the formation of some specific tissues and specific organs. So in the case of plant, we have initial result onto it have the very specific role into the lateral root formation. We call them LR development. So that is the basic function of this heme oxygenase into the plant system. However, in the case of some specific tissue, it has some specific role into the leaf, and then specific role into the root nodules, and then germinated seeds. So they are I just mentioned few of them. So in the case of leaf, I already mentioned that the phytochrome chromophore, this is the main role. Besides, the stomatal regulation is also controlled by the heme oxygenase. Then in case of root nodule, we have a rhizobium and the root interaction. So this enzyme, because this is related to the heme. So the lag hemoglobin, we know that the lag hemoglobin metabolism is all regulated by the heme oxygenase 1, HO1. And then in the case of germinating seed, it has some phycobilin synthesis role, and then inhibition of the seed germination, all that. So these are some specific role of these heme oxygenase into the tissues. Then one important, my collaborator, the Matthew Terry, which is working, he is working in Southampton. He mentioned this localization because in a very beginning slide, I have mentioned that this enzyme is present into the almost all the tissue, all the cells. So its localization is very important. So localization, it has the cellular cytoplasmic localization also, and then sometimes chloroplast localization also. So he mentioned, I specifically take this, Terry's paper slide, which is his focus is localization into the chloroplast, the stroma of the chloroplast is the one site of this localization. Besides the cytoplasm, obviously is present into the cytoplasm. So this is the one slide which mentioned its localization. When we design any experiment, so we have to start from the beginning, and beginning that means we have to check the role. If we standardize the role of any enzyme, any protein, any so then we do some fundamental experiment. Then what are the role of the heme oxygenation into the plant morphology, then physiology, and then molecular biology. So this is the design, and we cannot take the individual enzyme to check its activity. We have to do some another enzyme which is the same family and they also have the same function. So we regulate them. We need to control them in the different way, and then only can we take the new enzyme. So this is the one experimental design. So when we started the plant science, then we go for the hydroponic cultures. You can see the pictures of how we germinate the seed and how we establish the hydroponic culture. So we have 3-4 plant species on which we are working. One is the Brassica, then Glycine, then Cymopsis. After shifting the Jodhpur, I also added the Cymopsis, Tetragonaloba, and Eruca sativa to work on this. But initially, I started with the Brassica, the mustard, Indian mustard. So you can see the pictures of the Indian mustard, which is grow into the hydroponic cultures with some stress, because as an enzyme, we propose this enzyme as an antioxidant enzyme. So if any antioxidant is there, then we identify some of the stress and check its level. If it's increasing in somehow, then we have a basic idea that's helping the stress condition. So this enzyme could be act as an antioxidant. So to create a stress into the plant, we have the 2-3 methods. We can go by the abiotic stress or the biotic stress. So the whole work is the abiotic stress. So we are using with the salt stress and the metal stress. And now we have also added this temperature stress also. So these 3 stresses we have mentioned about the heme oxidase, and in these 3 stresses, we have calculated the enzyme concentration in the different tissue, different organ at the physiological level as well as the molecular level. So we will discuss all about this enzyme and its role. So these are the data, So we can, I don't discuss much about the data because you have the diverse audiences are there, because they have the different background, environmental science, ecology, and some other also. So just I mentioned the tolerance index. So we have to do some fundamental experiment related to the tolerance index. So we grow the plant in the hydroponic culture. One is the control plant, and rest is the treated plant, treated with the temperature, treated with the metal, and treated with the salt. So these are the parameters we have taken, and the different concentration of metal, different level of the temperature, and then salt we have taken. Then the control plant, we calculate the other enzyme, including the hemoxinase, and in the treated plant also we do the same. So you can see the morphological parameters. The root is the control plant is the longer one, and the treated plant is getting shorter. So that means the metal or the salt is having some negative impact on the morphology of the plant. They reduce the plant growth. This is the direct indication, and you can see into the picture. It started from the 10 micromole to we go for the 120 micromole of the metal, the cadmium metal, and the copper metal also. So we have the number of metals we have chosen. So I go for the random sliding here. So these are the metal we have chosen, and you can see the direct impact onto this. And some level there is an increase, decrease at the plant physiological parameter, and at the later age they have some increase also. So why this is happening? So that tolerance index, there is a formula, and we have also have an LWC formula. So we calculated with these formulas. Then we have this basic fundamental. Yes. So what are the difference between the control plant root length and the treated root plant of the root length? So where is the actually stress is caused? Initially, the plant growth is reduced very drastically, and after some concentration around 50 micromole, there is an increase into the root length again. So this is very important. So we call them lethal concentration and the sublethal concentration. Lethal concentration mean where the plant is get almost die, because that is the lethal concentration. But at this stage, you cannot calculate any enzyme, any protein, because plant is almost dying now. But the sublethal concentration, where the plant is struggling to survive, these are the perfect stage to identify the which enzyme is play important role to make the plant survive. So we call it the sublethal concentration. So in our case, the 50 to 75 micromole of the metal concentration are the sublethal concentration, where this plant is try to acclimatize with this new environment, the stress environment, and where we calculate the root length, shoot length, fresh weight, dry weight, and some enzymatic parameter as well. So you can see these pictures and how we calculate the stress. Plant cannot speak, I am in stress. So we have calculated some specific parameter. We call them MDA. The lipid peroxidation is very important. Once the lipid is get peroxidized, that means plant are in the problem. So we go for the MDA content analysis and then H2O2 content analysis. If the H2O2 is increasing, that means there's a toxicity is increasing. So these are the basic parameters through which we can calculate the plant stress. And then obviously, what are the enzymes they are upregulating in these stages? Not only the heme oxidase, there are a number of enzymes also which also help the plant. We call them ascorbate peroxidase, catalase, and peroxidase activity. Then APX activity. These are the other enzymes. They also play important role. But our focusing on the heme oxidase, so we directly jump onto the heme oxidase. These are the some other also enzymes with the same data. But you see, then one interesting other molecule, I have just one slide of this molecule, that is the nitric oxide. Interestingly, when we talk about the heme oxidase, the gaseous molecule, we call that the gaseous signaling molecule into the plant system, that is the nitric oxide, which is also accumulated in the root cells. And you see the this fluorescent microscopy of the treated plant and non-treated plant. If you see the slide, A is the control plant. There is no metal treatment, and the B, C, D, and these are the treated plant, where you can see the more fluorescence. And this green fluorescence indicates the production of the nitric oxide at the 75 micromole copper treated and cadmium treated brassica plants. So this is again, this molecule is also very important. Interestingly, this nitric oxide also having a very basic signaling molecule into the animal cells. So these both are having a common source, animal and plant, both present into both the category. This is again in the case of cadmium, and earlier it was in a copper. And this is the anatomy with the same, because I'm not go detailed part of the why this is happening. So anatomical level also we can check the presence of this nitric oxide. The more bluish color, that means the presence of the more nitric oxide. The lesser blue, that means that is the lesser amount of nitric oxide. But to capture it, we need some dye. So we have a specific dye. We call that NADPH diphorase activity. So through which we can have oxidation process, and we get this purple color. After having some basic idea of these enzymes and these parameters, we calculate the heme oxidase into the in all the metal cases as well as the NaCl cases with the same protocol. So here you can see the enzyme, the heme oxidase activity, and at the case of 70 micromole of the copper, cadmium, and almost 30 to 50 micromole NaCl, we get the maximum heme oxidase activity. So these are the yes, I just supposed to mention about especially for the scholars and students, it is not happen every time is heme oxidase is helping throughout the plant in some specific concentration only. Suppose at the 10 micromole, if the catalase is helping, then 50 at that 50 micromole at the heme oxidase is helping. This is something very fundamental of the plant physiology and molecular biology, because one molecule can't help throughout. One antioxidant can't help throughout the stress process. At some process, the proline is help. At some level, the catalase is help. At some level, heme oxidase is help. So this is some basic physiological phenomenon. And here you can see the heme oxidation activity in the case of cadmium. So at the 125 micromole of the cadmium, we get the maximum activity of the heme oxidase, almost at a 3.4 times higher, 3.45 folds higher activity. That means at this concentration, at a 125 for the cadmium and the 75 millimole of the NaCl. So these are the two concentration we have picked to go for the molecular study. So these are the all the physiological study. And you see this is the intracellular cadmium, where is the maximum amount of the metal and where is the maximum amount of the heme oxidase activity. So you can see these are the intracellular atomic absorption is the basic machine through which we can calculate the metal content into the plant tissues, into the leaves, shoot, and wherever you want. So these are the intracellular cadmium, which is corresponds to the heme oxidase activity. So this is the double check experiment. Again, you can see the various activity of heme oxidase after the. Then, I'm supposed to mention that there's a two type of experiment. One is the concentration dependent, and other are the time dependent. In one case, you have changing the time, but the concentration remain the constant. But at the other level, we have changing the concentration, but the time is the same. So this is how it's happening. So the two experiment, time dependent and the concentration dependent. So this is the time dependent. Is we have a time in hour of 30 minutes, then 6 hour, 15 hour, 24 hour, 48 hours, 72 hours, and 96 are the maximum limit. So this is the very interesting data which we have published into the Protoplasma. It's a very nice journal about plant physiology. So this is, and then this again this data with the different plant species in the case of cadmium. So you can see the heme oxidase activity, and the metal is directly the heme oxidase directly help into the this metal activity. So besides after screening of these basic sites or basic concentration and basic time, then we can check it's the molecular study. And the molecular study. So I directly jump onto the slide where we require. So we go for the total synthesis, cDNA synthesis process. We know that how this is happening because there's a standard protocol nowadays to have these all experiment I'm showing you that are the in case of Brassica argentea. So once we go for the cDNA synthesis, then we can go for its make it a double stranded DNA through the cDNA. So and then we go for the actin standardization. You will CR mean the control plant, and 50 micromole the cadmium root and 200 micromole of the cadmium root. So these two concentrations are chosen. And in the case of copper, we have chosen the one concentration only. So this is the actin standardization of the DNA. So what is the size of, because we need some ladder there. So this is the size of the this gene, that is the BJH, because this is the Brassica argentea, so we call the BJHO1, Brassica argentea hemoxinase 1. So you can see the semi-quantitative RT-PCR of the HO1 analysis in control and treated plant. So you see the control plant, there's almost no expression of the hemoxinase 1. But in the case of 75 micromole of the copper treated root and then cadmium treated root, you can see the clear visible of the gene. So this is the one aspect. Then we need to clone these gene. So we take this gene. I'll just directly jump to the this would be the more explanatory to all of you, because how we go for this analysis, because data is like more technical part of it. So besides we go for its physiological analysis, we can go for the semi-quantitative RT-PCR. And yes, this is the picture through which you can easily understand the whole process. So we ligate this gene in BJHO1 cDNA into the PGMT vector, which is very common and most common vector for this whole process. And then we transform this BJHO1 gene into the PGMT vector transport to the E. coli strain. And there are the number of E. coli strains through which we go for the cloning process. But in my case, I have go for the DH5 alpha. DH5 alpha is the commonly used E. coli cell for the transformation process. And then we go for the colony PCR, and you can see this picture. You might see my cursor. So 811 base pair gene you can see at the number 2 sites. And then we can check if the PGMT and HO together is the bigger molecule, and you can see after the digestion of this 811 base pairs of the almost gene it is. Then very important. You isolated the gene, but what is the sequence of the gene? That is we need to know. And for this we can go the sequence analysis. And in the case of Brassica argentea, we have sequenced this gene with the PGMT vector. So because PGMT is the commercial vector, and we know it's a sequence, and we sequence it with the PGMT, and now we know the sequence of this HO1 gene, which is a bit new in the terms of we know first time. Otherwise, obviously, it's already present into the plant species. So you can see these are the accession number which we have submitted in the NCBI JX27. If you check it, you can check with the Sekhaut and Dixit. Subham Dixit is my student. She is also worked during her PhD program. So it's already submitted to the NCBI. Then because we need to know it's a protein, not on the gene, because gene is there, but what type protein it is, the protein structure is required, the how its enzyme work is, that is required. So we elute, we cut this gene from the PGMT vector, and now we use another vector, that is the PET vector. We call that transformation vector, PET28, PET28 vector. So we put this into the PET vector and then again transfer to the E. coli cells. Now we have another E. coli cell that we call the BL21 cells. These are the engineered cells through which we can translate the whole any gene. So these are the common one. Besides, there are number of cells are available, depends on to their experiment and gene type, gene size. So you can go for that. So you can put the gene into the this PET vector, and you can see the colonary PCR with the PET plus HO1. Earlier it was the PGMT plus HO1. Now it's the PET plus HO1. So we have the number of colonies we have run into the gel, but ultimately we check is the digestion with the to remove the PET vector. We digest with the NOT1 and SAC1. These are enzymes which remove the gene, and you can see this is the Western blot, and this is the 1100 base pair of the, we can isolate it, and then we culture the positive colon in IPTG. Most of the people, those who are working with the molecular aspects, they know what is the IPTG. This is to enhance the expression of the any gene into the cultures. So these are the, you can in the lower portion of the this screen, you can see the SDS phase, and then this induced HO1 induced gene is now HO1 protein. Earlier it was the gene, now it's an HO1 protein. So the size is almost 32 kD of the size, and you can see the Western blot of this this gene. So this is the novel gene which is having a very specific role into the plant stress tolerance mechanism or endurance mechanism. So this is fundamental aspect of it. Now we can go for its in a location, because in a very beginning slide I mentioned that the localization of this gene is very important, and Matthew Terry has mentioned its location into the chloroplast. So we have chosen both the organelles, mitochondria and chloroplast together, and the chloroplast is the most common site because it's related to the chlorophyll. So we isolate these both of the organelle and check with its localization into the chloroplast and the mitochondria. So earlier slide might be the bit more visible. So this is the semi-quantitative RT-PCR of the chloroplast DNA, cpDNA, and mtDNA. So here you can see the cpDNA, that the chloroplast DNA is the more shiny band you can see. That means this is the confirmation of that localization into the chloroplast sites. So obviously the this base pairs are a bit vary than the cytoplasmic DNA. Then Western blot analysis also did into the chloroplast and mitochondria. In the case of chloroplast and mitochondria, almost they are the same size of the protein, that is the 30 kDa kilodalton. So this is the size of the protein, and we have is the crystallography in all data which is not be received yet. So we are trying to go for the 3D structure as well. So in summary, you can see how the heme oxidase is working in the case of plant species. So if any type of the stress, which is we have gone through that the three stresses, salt, metal, and the temperature. I didn't show any temperature data in this presentation because they are not completed yet. So in the case of stress, the heme is degraded in the case of heme oxidase, and this heme is in the three way. There's a three component is released by the heme oxidase. Heme oxidase 1. That is the carbon monoxide, iron, and biliverdin. So these all three, all three degradation part of the heme is used for the cell protection in one way or other way. But yeah, we know the role of carbon monoxide in animal. There's a lot of study about the carbon monoxide, and the biliverdin is also play a positive role in the cell protection. Iron is have some data is in the favor of the iron, and some data is not in the favor of the iron, but still it's there and it's helped somehow. So one can be explore the how this released iron is play role into the cell protection. But this is in our case also we have gone through this data, and then carbon monoxide and the biliverdin is a role in the cell protection. So hemoxy individually play important role in the cell protection. However, the produce is also play important role to the cell protection. And this is the whole picture of the heme metabolism, how it is happening, what are the synthesis process, what is the precursor. So in the side, in another side of the slide, you can see the whole process of the heme metabolism. So this paper we have long back published in the Journal of Experimental Botany. Now we can summarize the whole process into another slide, that is the lipid peroxidation and the gene regulation. So these are the some very important molecule which is play important role during this process. That is the proline, ascorbic acid, carotenoid, polyphenols, antioxidant enzymes, and thiol components. They all play in the cell protection. However, the HO is directly involved into this process. That is we have focused the whole story about the heme oxygenase. So the earlier presentation by the CP Singh, he has mentioned a lot about the microRNA, and these RNAs are very important. And if any microRNA regulate the enzymes activity and some other components activity, and that is very new trend into the any animal science research or plant science research and bacterial research. So how we can check the microRNA, which is directly involved in the process of control the any of the enzyme activity or any other components activity. So we have mentioned, not go to detail of this because it's already in the last presentation is explained very nicely the role of microRNA, and it's it's a process. So this is the but I have just mentioned the role microRNA related to the heme oxygenase, just the one slide. So this is the comparative chart of the animal microRNA and the plant microRNA. So we know that different microRNA are present in the case of animal, because the animal is all to the different cell ambience they have. But in the case of plant system, the microRNA basically they have an ARFs, oxygen response factors. So they these ARFs is directly involved into the HO regulation. So oxygen response factor is regulated by the by HO1, and in the both together they go for the developmental aspect of the this is help into the LR formation. So this is the one aspect of the mRNA which is regulated by the ARFs. Then in case of another HO1 regulation, so ARF is there, so oxygen ARF, oxygen response factors. So oxygen have regulate the CDKs, and this cyclic protein is directly involved into the this regulation of the heme oxygenase, which is further help into the LR formation. So this is a very another important aspect of the heme oxygenase into the plant cell, which is it's almost new into the cell, and any scholar can pick this particular topic to explore into the different. If you do any aspect of the heme oxygenase to the plant, it would be the new one, and you get a very nice data as well as the nice publications. So I've summarized this whole story here only, that with the recommendation, the heme oxygenase is the new molecule into the plant system, and very few labs are working with this enzyme. And these are the some publications related to the heme oxygenase. Anybody can take these from the internet. If anybody have need the how to calculate, how to estimate this enzyme into this, anybody can go for the microbes also, because there's a very less study into in the case of microbial system. So they can only need to modify the protocol to estimate or analysis of the heme oxygenase. So anybody can take this up. Yeah, this is the another very summarized form which is recently come into the this year only. So the role of heme oxygenase into the lateral root formation, phytochrome chromophore synthesis, HO1 signaling, antioxidant mechanism. This the old data is summarized into this plant cell environment journal, which is published this year only. So this is also free available onto the Google. Anybody can take this. by the name of the heme oxidase, a functional diverse enzyme. So I must acknowledge the many people, the Professor Batra, Donald Haney, DST Boycott Fellowship, Insight International Fellowship, Matthew Terry, my collaborator, and the Karina Balestresi from Argentina. They are the collaborator of mine this the whole study. So I have the one part of the study I have discussed with you all. These are the research groups. We have different labs, because I have this data is collected from the 2-3 labs. So I must acknowledge them all. You can see the pictures of the. So thank you very much you all for listening me patiently, because I have a 15-minute slot. So I don't think I've taken much. Thank you very much. If you have any query, you can ask me. I'll be happy to interact with you all.
Thank you so much, sir, for your inspiring words. Your presence and wise words help magnify our cause in the best possible way. We thank you, sir, for being with us today.
Thank you very much.
Next, I would like to invite Rajita Patni from Indira Gandhi Center for HEAPS for her presentation.
Hello.
Yes, Rajita, you can.
I would like to share my screen. Okay.
Rajita, you can share your screen now.
Namaskar everyone. My name is Rajita Patni. I am from University of Rajasthan, and my topic is textile dye degradation by nanoparticles. Why nanoparticles? Because they have unique physical and chemical properties from their known materials in bulk because of their size. They have high reactivity and large specific surface area. They can be efficiently used as solutions to a wide range of environmental problems we face nowadays. When we talk about textile effluents, they are killing rivers worldwide because there are certain industries which do not
Rajita, we are not getting your voice. We are not getting your voice, Rajita. You're not audible. Now I would like to invite our next presenter, CONIAPS-XXVII from Indira Gandhi Center for Human Ecology, Environment and Population Studies, University of Rajasthan, Jaipur.
Hello everyone, respected chairperson, convener and director of the organization. Myself Jakrit Singh, and I am a research scholar from IGC for HIPS, University of Rajasthan. My topic for presentation is human-wildlife conflict in Rajasthan. Next slide, please. Human-wildlife conflict can be defined as any negative interaction between wild animals and human beings with undesirable consequences for both people and their resources, and wildlife and their habitat. Human-wildlife conflict occurs when animals pose a direct and recurring threat to livelihood or safety of people. This is not a new thing. In ancient times, people used to hunt wild animals for their own protection or protection of their resources. But nowadays, the increased frequency of wildlife and human conflicts is due to the deforestation and destruction of their habitats. Next slide, please. There are many reasons for conflicts between the human and wildlife. Among these, major are the increase in human population, deforestation, loss of habitat of wild animals, climate change, food availability, and construction of roads through the forest. As we can see in the graph, in 1971, Rajasthan has a population of 25.8 million, while in 2011, it is 68.6 million. Next slide. While we can see, because of our conservation efforts, the number of leopards in Rajasthan are number of tigers in census report 2018 has been increased from 32 in 2006 to 69 in 2018. That is an average number. While the forest cover in Rajasthan is only 34,224 kilometers square. That is only 8% of the total geographical area of Rajasthan, while in this only 0.02% area is dense forest, while others are open forest or light forest. So wildlife animals faces great threats from humans, or this in search of food, they move to another regions. The reserved and the protected and unclassed forests are 38.11%, 55.64%, and 6.25% respectively in recorded forest area in the state, according to the Indian Forest Survey of India 2019 second volume report. However, as the digital boundary of recorded forest area from states cover only 33,072 square kilometers. The livelihood of the local communities of Rajasthan are primarily based on the rearing of livestock. In India, the state stands second in the number of heads of livestock, that is 577.32 lakhs, while it stands seventh with a human population of 744.88 lakhs. It is also a common reason for conflict between wildlife and humans, as the wild animals graze in the reserved forest. Most sanctuaries in the state face enormous biotic pressure due to livestock grazing, while the livestock killing by big cats is considered to be a major issue relevant to rural livelihood and conservation, because many carnivore species have been heavily prosecuted as a direct result of elevated conflicts level with communities. In general, people use physical barriers to protect their resources from wild animals, such as fencing, wiring, and other type of cloth covering for their crops. While the translocation programs are for people or villages, those fall in the region of the reserved area. Compensation policy is also a mitigation method, but it is very less in Rajasthan. We need to implement more advanced and technology-based solutions to avoid conflicts, such as use of blinking lights or paintings, while increasing awareness or translocation programs for various wild animals, afforestation.
On the behalf of entire team of Indira Gandhi Center for Human Ecology, Environment and Population Studies, I would like to thank Subhash Chandra sir for taking time from his busy schedule and being the chairperson at our conference. All your research work has inspired so many people, sir. Thank you so much for being with us, sir.
Thank you. And
Now
hello.
I would like to invite Dr. Surendra Singh Chauhan, sir, Director and Convener of IGC for NEEPS, for the presentation of the conference report.
Good evening. Good evening, everyone. Professor Subhash Chandra of Ajmer University, Professor Naresh Malik, Professor Pankaj Kumar Jain, Dr. Abhimanyu Singh, distinguished guests, faculty members, colleagues, participants, ladies and gentlemen. On behalf of Indira Gandhi Center for Human Ecology, Environment and Population Studies, University of Rajasthan, Jaipur, and International Academy of Physical Sciences, Prayagraj, India, I would like to thank you all for attending this 3-day international conference on ecosystem restoration and environmental sustainability concerns and alternatives. We hope that you all found the conference informative and worthwhile.
The primary goal of this conference was to bring together highly educated and well-versed professors as well as inquisitive researchers and students from all around the world on a single platform to discuss the issue we are facing regarding environmental degradation and to develop possible strategies and alternatives to eliminate these problems. We believe that our diverse and dynamic group of speakers and panelists provide in-depth insight as well as actionable and practical solutions. The conference was inaugurated on 26 October 2021 by Professor B.L. Sharma, former Vice Chancellor, University of Rajasthan, Jaipur, and delivered the keynote speech. Professor Rajiv Jain, Vice Chancellor, University of Rajasthan, Jaipur, presided over the program. Dr. Pankaj Kumar Jain, Organizing Secretary, gave vote of thanks. Professor V.K. Mattu of Simla University, Himachal Pradesh, gave intuitive and informative speech on apiculture. He delivered this informative speech on the use of honey, royal jelly, and various products which can be get from apiculture, and also gave information about the use of insecticides and their harmful effects. Professor M.M. Ranga of Sarguja University, Chhattisgarh, delivered the lecture on environmental restoration and sustainable development goals, ecosystem services. Our first technical session was chaired by Professor Sandeep Arya from Institute of Environment and Development Studies, Bundelkhand University, Jhansi, in which Dr. Sharda Devi, Dr. Chetna Pradhan, Prasanjit Patari, Sanjay De, Uttam Nath, and Jyoti Vaishnav gave their oral presentation. Our second technical session was chaired by Professor Prahlad Dubey, Department of Zoology, University of Kota, in which Aradhana Chauhan, Bisab Nath, Supriya Adhikari, Sneha S, Hitesh Parikh, and Uruj Shabir presented their work. On 27th October 2021, Professor Anil Shrestha, Chairperson, Department of Viticulture and Technology, California State University, gave an enlightening lecture on pesticide use, its effects on agriculture, environment, and human health. Professor Neelam Mattu of Shimla University, Himachal Pradesh, delivered her speech on honey and honey bees. She shared various phrases from Upanishad and Vedas. Later in the day, Dr. Ranveer Singh, University of South Korea, shared informative knowledge about solar energy and solar panels, followed by the session on regulating the subscription economy to ensure sustainable consumption and production pattern, 12th goal of sustainable development for restoration of ecosystem and environment by Professor Rajiv Sijaria of Atal Bihari Vajpayee School of Management and Entrepreneurship, JNU, New Delhi. Our third technical session was chaired by Professor S.B. Atade of North Maharashtra University, Jalgaon, in which Shambhul Rahman, Namrata Singh, Subarto Ghosh, Elbahi A, Gyan Prakash Sharma, Canadian R, Iti Gupta, Petrai Datta, Pooja Rani Saha, Rudrom Babina Devi, Mohammad Safir, Ashok Kumar, Sutradhari, Arun Kumar Paul, Ramendra Soni, Nasreen Amin, Satyab Sahi Basu presented their papers. Today, that is 28th October 2021, Professor N.P. Singh, Department of Zoology, University of Rajasthan, Jaipur, delivered his talk on diversity of spider species and their role in the ecosystem. Our second guest, Professor S.C. Joshi, Department of Zoology, University of Rajasthan, Jaipur, explained how Indian spices can prevent heart disease and related risk factors. Our third guest, Professor Gyan Singh Shekhawat of Jai Narayan Vyas University, Jodhpur, gave speech on hemoxygenase 1 in modulating antioxidant defense response to oxidative stress. Our technical session was chaired by Professor Subhash Chandra of MDS University, Ajmer, in which Margi Patel, Sweta Singh, Jyoti Sharma, Tanisha Kataria, Deepan Sharma, Dr. Najia A. Rashidi, Sushila, Nidhi Rajoria, Suman Paul, Dr. Manvi, Malwal Sneha S, Ariban Devi Das Sharma, Devashish Sahu, Ms. Manju, Subhas Chandra Arora,
Biswajit, B. Shivani Sharma, Harsha Parwani, Dr. Chandra Pol Singh, Garima Singh, Rajita Patni, Jackrit Singh, Shivani Sharma presented their papers. Once again, on behalf of Indira Gandhi Center and on my own behalf, I thank you all. Thank you very much.
Now I would like to invite Dr. Pankaj Kumar Jain, Organizing Secretary of the conference, for the vote of thanks.
Good evening to all. Honorable Professor G.S. Segawa sir, Jainarayan Vyas University, Jodhpur. Dr. Surendra Singh Chauhan sir, convener of the conference and director of Indira Gandhi Center for Health. Professor Subhash Chandra, Head Department of Zoology, MDS University, Ajmer. Professor Naresh Mali, Department of Geography, University of Rajasthan. Dr. Chagan Lal, Department of Physics. Dr. Rama Solomon, Indira Gandhi Center for Hips. Dr. Abhimanyu, distinguished guests, participants, and students. I feel honored and privileged to get the opportunity to propose a vote of thanks on the valedictory function of International Conference on Ecosystem Restoration and Environmental Sustainability, Concern and Alternatives on the behalf of the Indira Gandhi Center for Hips, University of Rajasthan, Jaipur. I sincerely thanks to our chief guest, Professor B.L. Sharma, sir, that he humbly accept our invitation and delivered a keynote speech, and Honorable Vice Chancellor, Professor Rajiv Jain, University of Rajasthan, for presiding the function and gracing the inaugural function I am also thankful of the Professor G.S. Shekhawat sir for accepting invitation to be as the chief guest of the valedictory function. I sincerely thanks to all our invited lecturers to deliver a very informative talk and enlightening all the participants. I am also thankful to the President and Secretary of the IAPS, Priyagraj, to give the opportunity and select us as a host for the conference. I thank to all the plenary speakers, tutorial speakers, and other delegates for their enthusiastic participation in the conference. I am thankful for the university authorities for providing all facilities related to the conference. I am also thankful to all our research scholars, especially Rajita Patni, Jackrit Singh, Shivani Sharma, Jyoti Vaishnav, and Garima Singh, who have greatly helped us in organizing this seminar. I am also thankful to Mr. Rakesh Kumawat and Mr. Ravi Shankar Sharma for the technical assistance, and live telecast of the conference. I am also thankful to Mr. Pawan, TCO of the Indira Gandhi Center, for preparing the abstract book of our conference. Time does not permit to thanks to all by the name. So I want to thanks all the persons who help us directly or indirect way. In the end, thanks you all for being with us today. Have a wonderful day ahead. Thank you, Jay.
Congratulations to entire team and all the organizing members, convener, organizing secretary, and department also. Congratulations.
I would like to thank all the participants and everyone who has joined us for the conference. We are sorry for any inconvenience caused in the conference. Feedback links related to the conference will be shared on the Telegram group, and all the details regarding the e-certificates will also be posted in the Telegram group. Thank you, everyone.